Hello and welcome. In this calm and quiet journey, we will explore the wonders of biology together. Make yourself comfortable, breathe softly, and allow your mind to float gently. We will wander through the fundamental topics of life science, from the tiniest building blocks of matter to the harmony of entire living systems. This journey will be slow and soothing, like a bedtime story woven with scientific whispers. There is no need to grasp everything at once. Just let the words wash over you in a peaceful drift. As you listen, you can simply relax and let the gentle narration guide you through the world of biology. We begin at the very foundation of life and gradually build our way up, step by gentle step, reinforcing key ideas in a soft and reassuring way. So settle in and allow yourself to be carried on this calm exploration of life. Together, we will delve into the basics of biochemistry, the large molecules of life, the structure of the cell, the flow of energy in metabolism, respiration and photosynthesis, the genetic code, and the systems that keep the body in balance. All of this will unfold slowly with soft transitions from one idea to the next. If your mind wanders or grows drowsy, that's perfectly fine. Just know that the essence of these ideas will surround you and comfort you. Now, let us gently begin our journey into the world of biology. Biochemistry, the chemical foundation of life. In the beginning, everything about life comes down to chemistry. Our journey starts with the smallest pieces of matter, atoms. Then atoms join together through bonds to form all the substances around us, including the components of our own bodies. Some bonds are strong and tight, while others are gentle and easily broken. When two atoms share electrons between them, they form a covalent bond, holding together like two friends holding hands. If the sharing is equal, the bond is non-polar. And if one atom holds the electrons a bit closer, the bond becomes polar with a subtle electrical difference between the two atoms. In other cases, one atom can completely take an electron from another. This creates charged atoms that stick together like tiny magnets. That attraction is called an ionic bond, and it happens, for example, when sodium and chlorine combine to form the salt crystals in the ocean. These different bonds, covalent and ionic, are the threads that weave molecules together. Even between separate molecules, there are gentle forces of attraction called intermolecular forces. The weakest of these are van der Waals or London forces, which are faint touches between all atoms and molecules. Stronger than those are dipole-dipole forces, which occur between polar molecules that have slight positive and negative ends. And strongest of all, though still much weaker than covalent bonds, are hydrogen bonds, special attractions where a hydrogen atom in one molecule is drawn toward a very electronegative atom like oxygen or nitrogen in a neighboring molecule. These hydrogen bonds are quite magical in biology, as we will see, because they give water and many biological molecules their unique properties. Now imagine a droplet of water. Water is simple, just two hydrogen atoms bonded to an oxygen atom, yet it is profoundly special. Water molecules love to stick gently to each other through those hydrogen bonds, making water wonderfully cohesive. This cohesion is why water forms round droplets and why it can travel up the thin vessels of a plant from roots to leaves. Water is often called the universal. So many substances dissolve in it. It's as if water can welcome many different molecules into its embrace. The oxygen in water carries a slight negative charge, and the hydrogens carry slight positives, so water can surround charged ions or other polar molecules and carry them into solution. This property makes water an ideal medium for the chemistry of life. Inside our cells, countless reactions occur in an aqueous, watery environment. Water also has a high heat capacity, meaning it can absorb a lot of heat energy with only a small change in its own temperature. Because of this, water warms up and cools down slowly, helping to stabilize temperatures in our bodies and in the environment. Think of the ocean staying cool in summer and warm in winter. Water gently buffers the world from extreme changes. When water evaporates or turns to vapor, it takes away a great deal of heat, high heat of vaporization, which is why a breeze on damp skin feels cooling. Another almost magical trait, water becomes less dense as a solid than as a liquid. Normally you might expect a solid to sink in its liquid form, but ice floats upon water. This is because the hydrogen bonds spread water molecules into a lattice when frozen, making ice expand and become lighter per unit volume. On a winter pond, ice forms on the surface and floats, gently insulating the liquid water and the life swimming below from the freezing air above. In this way, water protects life even in cold conditions. All these properties, cohesion, solventability, heat buffering, and floating ice, show how perfectly designed water is to cradle life. Within water and other solutions, we often find substances called acids and bases. These are like two opposite ends of a spectrum of chemical nature. An acid is a substance that, when dissolved in water, releases hydrogen ions H-, essentially increasing the amount of free H- in the solution. A familiar example is lemon juice or vinegar, which are acidic and have that sour taste. A base, on the other hand, reduces the concentration of hydrogen ions, often by releasing hydroxide ions, OH- that can capture H- and remove them. Soap or baking soda are basic substances. We measure how acidic or basic a solution is using the pH scale. This scale runs from 0 to 14. In the middle, pH 7 is neutral. Pure water is around pH 7. If a solution has a pH less than acidic, more H-, and if greater than 7, it's basic, more OH means. It's a logarithmic scale, which means that a drop of 1 pH unit, say from pH 7 to pH 6, actually means the H- concentration is 10 times higher. Each step is a 10-fold change. So a pH of 4 is not just a bit more acidic than pH 7, it's 1,000 times more acidic, because 7 to 4 is 3 steps, 10, 10, 10. Our blood is slightly basic around pH 7, 4, and it's important that it stays in a narrow range. To keep solutions like our blood from getting too acidic or too basic, the body uses buffers. A buffer is a substance or a combination of substances that resist changes in pH. Buffers can soak up excess H- ions when things get too acidic, or release H- when things become too basic. They act like a gentle guardian, keeping the environment stable. This way, even if metabolism or other processes produce acids or bases, the overall pH of our cells and blood remains steady, which is vital for the proper function of enzymes and other molecules. Speaking of molecules, let's recall one very special element: carbon. Carbon is often called the backbone of life. Almost all the chemicals in living organisms are built on carbon atoms. What makes carbon so extraordinary? Carbon atoms have 4 valence electrons, which means they can form 4 covalent bonds with other atoms. This allows carbon to act like a versatile connector, bonding with up to 4 other atoms at once. A single carbon atom can bind to other carbons, forming long chains or rings, and at the same time bind to elements like hydrogen, oxygen, nitrogen, and more. Because of this, carbon can form a tremendous variety of structures. Imagine a rich assortment of shapes: straight chains, branching trees, rings, cages, all possible with carbon. Hydrocarbons are the simplest carbon compounds, containing only carbon and hydrogen. They can be gases, liquids, or solids and form the basis of many fuels and natural oils. But in biology, carbon usually comes adorned with other atoms or groups that give molecules specific characteristics. These attachments are known as functional groups. For example, an OH hydroxyl group makes a molecule an alcohol, a MyCOH carboxyl group makes it an acid, an NH2 amino group makes it a base, and so on. These functional groups are like personality traits on a carbon backbone, affecting how a molecule behaves in water or in reactions. Let's imagine all these tiny chemical pieces coming together gently. We have atoms forming bonds to become molecules. We have water providing a nurturing environment. We have acids and bases dancing around, and buffers ensuring everything stays in balance. This is the quiet chemical symphony underlying life. A biochemistry that operates all around and within us, even as we rest. With this foundation of basic chemistry, we are ready to see how these small pieces assemble into larger pieces. Tiny molecules will link up to form bigger structures that are essential for life. With a slow, deep breath, let's move from simple chemistry to the marvelous macromolecules of life, the large molecules that make up our cells and bodies. Macromolecules, the molecules of life. From simple building blocks, life constructs grand structures. Many small molecules, monomers, can join together like beads on a string to form a polymer, which is a long-chain molecule. These large molecules are often called macromolecules, and they truly are the materials of life. Imagine monomers as individual Lego bricks and polymers as the long chains or complex models you can build from those bricks. Life primarily uses 4 kinds of macromolecules: carbohydrates, lipids, proteins, and nucleic acids. Each type has a unique role in the cell, and each is made by linking smaller units into a bigger unit through gentle chemical reactions that often involve removing or adding water, dehydration, or hydrolysis. Let's explore each of these macromolecules in turn, softly and clearly. Carbohydrates, sugars and structure. Carbohydrates are sugars and starches, molecules that often taste sweet or store energy. The simplest carbohydrates are single sugar molecules called monosaccharides. A classic example is glucose, a sugar with 6 carbon atoms and with the formula C(CH)O6, which is a primary fuel for cells. Glucose is small and sweet, dissolves in water, and can be absorbed easily by our bodies to give quick energy. Monosaccharides typically have carbon atoms arranged in a ring, and they can have slight variations. Some have an aldehyde group, we call these aldoses, and some a ketone group, ketoses. But those are technical names for where a certain double-bonded oxygen is located on the molecule. The important thing to remember is that these simple sugars are the building blocks. When 2 simple sugar units bond together, they form a disaccharide, a double sugar. For instance, table sugar, sucrose, is a disaccharide made of glucose and fructose linked together. A disaccharide is like 2 rings joined hand in hand. These can still be sweet and easily broken down. When you eat sucrose, your body will break it back into glucose and fructose. Now, if many sugar units link up, one after another, we get polysaccharides, which are complex carbohydrates. These can be very large, sometimes containing thousands of sugar units. They usually are not sweet and often are not soluble in water. Instead, they serve as energy storage or structural materials. For example, starch is a polysaccharide made by plants to store energy. When a plant has extra glucose from photosynthesis, it hooks them together into starch, which is like a big spiral or branching chain of glucose units. When you eat a starchy food like rice or potatoes, your digestive system gently breaks that starch back down into glucose units, which your cells can then use for energy. Animals, including humans, store glucose in a similar polysaccharide called glycogen, which is stored in our liver and muscles for short-term energy reserves. Glycogen is like a highly branched tree of glucose units, so it can release glucose quickly when energy is needed. Polysaccharides can also provide structure. Plants, for instance, build their cell walls out of cellulose, which is a long polysaccharide of glucose, but linked in a way that gives it great strength. Glucose units in cellulose are differently from those in starch. Cellulose is tough and fibrous. It's what wood and paper are mostly made of, and most animals cannot digest it, though some, like cows and termites, have microbes that help them break it down. Still, cellulose fiber is important in our diet to keep our digestion healthy, even if we don't get energy from it. Another structural polysaccharide is chitin, which forms the exoskeleton of insects and crustaceans and the cell walls of fungi. Chitin is like a modified carbohydrate, where the sugar units have a bit of nitrogen-containing decoration. Imagine the crisp shell of a beetle's back or the crunch of a shrimp's tail. That's chitin, providing a hard protective structure while still being made from sugar subunits. So, carbohydrates can be sweet sugars fueling our cells or strong fibers supporting organisms. They range from a single ring, glucose, to gigantic polysaccharide chains. They are formed by joining monomers with the release of water in a dehydration synthesis reaction, and can be broken apart by adding water back, hydrolysis, a gentle back and forth dance of chemistry that allows energy to be stored and released as needed. In the body, enzymes oversee this process, making sure it happens under the mild conditions of living cells. We'll talk more about enzymes soon, but let's continue our exploration of macromolecules. Lipids, fats and membranes. Now we turn to lipids, which include fats, oils, and related molecules. Think of lipids as the hydrophobic, water-fearing molecules. They do not mix with water easily. If you drop oil into water, it will separate and float, forming little shimmering bubbles, because oil molecules want to clump together away from water. This is due to their largely non-polar nature. Lipids are made mostly of carbon and hydrogen, with very few oxygen atoms compared to carbohydrates, so they are mostly non-polar hydrocarbons. Because of this, lipids are insoluble in water, but soluble in other non-polar solvents, like dissolves like, as the saying goes. The most familiar lipids are fats and oils, also known scientifically as triglycerides. A fat molecule is made from a glycerol, a small 3-carbon backbone, attached to 3 fatty acid chains, hence the name triglyceride, 3 fatty acids. Fatty acids are long chains of carbon and hydrogen. If every carbon is holding as many hydrogens as possible, we call it saturated fat, found in things like butter, solid at room temperature. If there are double bonds between some carbons, then it is unsaturated, found in oils like olive oil, liquid at room temperature. The double bonds create kinks in the fatty acid chain, preventing the molecules from packing tightly, hence unsaturated fats stay fluid. Fats are nature's way of storing energy in a compact form. Lipids carry more than twice the energy per gram as carbohydrates do. That is why animals use fat for long-term energy storage. It's very energy dense. Picture a bear preparing for hibernation, eating lots of food to build up fat reserves. The fat will sustain it through the long winter because of all the energy packed in each molecule. In our bodies, fat not only stores energy, but also provides insulation, keeping us warm and cushioning for organs. Another important group of lipids are phospholipids. These are special because they are dual-natured. Part of the molecule loves water, hydrophilic, and part hates water, hydrophobic. A phospholipid is similar to a fat, but instead of 3 fatty acids on glycerol, it has 2 fatty acids and 1 phosphate group with additional small polar group attached. This gives a phospholipid a polar head, the phosphate-containing part, and 2 non-polar tails, the fatty acids. In water, phospholipids arrange themselves such that the heads face the water and the tails hide away from water. This property is the foundation of cell membranes. In cells, phospholipids form a bilayer, 2 layers of molecules with tails inward and heads outward, creating a delicate yet dynamic phospholipid bilayer membrane that encircles the cell and many of its internal organelles. This membrane keeps the inside of the cell separate from the outside, like a soft boundary, but because of the fluid nature of lipids, it remains flexible and not brittle. We will talk more about cell membranes in a little while, but it's amazing how self-organizing these phospholipids are. Simply by their love and fear of water, they spontaneously form bilayers when in an aqueous environment, forming the very container of life. Lipids also include steroids, which have a very different structure. They are made of 4 fused carbon rings. Even though they look different from fats, they are also hydrophobic. Cholesterol is a steroid that is an important part of animal cell membranes, helping to maintain fluidity. Cholesterol is also the starting molecule for many hormones like estrogen and testosterone, which we will meet again when we discuss the endocrine system. So even though steroids are lipids, they often act as signals or structural components rather than energy storage. Then there are waxes, another type of lipid. Waxes consist of long chains of hydrocarbons and are solid at room temperature. Plants often use wax to coat their leaves and fruits, creating a waterproof barrier that prevents water loss. Think of the glossy surface of an apple or the sheen on a holly leaf. Bees construct their honeycombs from beeswax, which is yet another example, a sturdy but pliable material made by linking fatty acid chains to long-chain alcohols. Waxes are incredibly water repellent. They ensure that water rolls off surfaces. Finally, pigments like carotenoids are also considered lipids. Carotenoids are the orange and yellow pigments found in carrots, pumpkins, and autumn leaves. They have long chains with alternating double bonds, making them coloured, and they dissolve in fats, which is why, for example, beta-carotene, the orange pigment in carrots, is best absorbed if you eat carrots with a little fat. These molecules, being lipids, also reside in our cell membranes and can play roles in capturing light in plants or as antioxidants. In essence, lipids provide energy storage, make up membranes, serve as waterproof coatings, and even act as hormones and pigments. They are diverse in form yet unified by their hydrophobic character. They float calmly in our story as the silky, non-mixing counterparts to the water in our bodies. Proteins, workers and building blocks. Moving on, we arrive at proteins. perhaps the most versatile and complex macromolecules. If we think of a cell as a bustling city, proteins are the workers, the machines, and sometimes the building scaffolds of that city. Proteins are made of smaller subunits called amino acids, strung together in chains. There are 20 different amino acids commonly found in proteins, and each one has a common structure with an amino group, an NH2 on one end, and a carboxyl group, COOH, on the other. Because of these groups, in water, an amino acid can act both as an acid and a base, giving it some interesting properties. But the part that differs between amino acids is called the R group or side chain. It could be as simple as one hydrogen, as in glycine, the simplest amino acid, or a more complex ring structure. Some side chains are non-polar, oily. Some are polar or charged, like acids or bases themselves. This variety in side chains is what gives proteins their complex personalities. Amino acids join together by peptide bonds, a bond between the carboxyl group of one amino acid and the amino group of the next, with a molecule of water released in the process, another dehydration synthesis. When two amino acids link, you get a dipeptide. When many link, you get a polypeptide. A protein may consist of one or more polypeptide chains folded and coiled into a specific shape. It's important to note that the sequence of amino acids in a protein, its primary structure, determines everything about the protein's final shape and function. The sequence is like a long word spelled out in a 20-letter alphabet, each letter being an amino acid. This sequence is dictated by DNA, which we will discuss later. Once the chain is formed, it doesn't stay as a simple line. It starts to fold up. Portions of the chain may coil into a spiral called an alpha helix, or fold into a pleated sheet, and these are known as secondary structures held together by hydrogen bonds between backbone atoms in the chain. Not all proteins have these structures, but many do. Then, the entire polypeptide assumes a tertiary structure, a complex 3D shape formed by interactions between the side chains, R groups, and the backbone. These interactions can include hydrogen bonds, ionic bonds, hydrophobic interactions, nonpolar side chains clumping inside away from water, and even disulfide bridges, strong covalent bonds between sulfur-containing cysteine amino acids. The tertiary structure is critical. It's what makes one enzyme specific to its substrate or one antibody specific to its antigen, for example. Some proteins stop at this single-chain tertiary stage. Others have more than one polypeptide chain and thus a quaternary structure, where multiple folded chains fit together to form a functional unit. Hemoglobin, the oxygen-carrying protein in our blood, has 4 subunits that come together, for instance. You can picture a protein as a delicate origami or a knotted ribbon. If the shape is lost, the protein often loses its function. This can happen by denaturation, where a change in temperature, pH, or chemical environment causes the protein to unfold or misfold. For example, if you cook an egg, the heat causes egg white proteins to denature and solidify. The process is irreversible in that case. The clear raw egg white becomes opaque and white. In the body, a high fever can start to denature proteins, which is dangerous. However, under normal conditions, most proteins maintain their proper shape, or if they misfold, there are cellular helpers called chaperones that assist them in refolding. Occasionally, some small proteins can renature, refold back, if the normal conditions are restored, but many cannot, which is why homeostasis, keeping conditions like temperature and pH stable, is so important, a topic we'll revisit later. Proteins are truly the workhorses of the cell. Enzymes are proteins that act as catalysts, speeding up chemical reactions without being consumed. They lower the activation energy needed for reactions to proceed, which means reactions that would be too slow to sustain life can happen in fractions of a second with an enzyme's help. For instance, the enzyme catalase, which is found in our liver, helps decompose hydrogen peroxide, a harmful byproduct of metabolism, into water and oxygen gas unbelievably fast, millions of molecules per second. Without catalase, that reaction would be too slow and peroxide would accumulate and damage cells. We will explore enzymes in more depth shortly because they deserve their own gentle discussion. Proteins also form structures like collagen in skin and connective tissues, giving stretch and strength, keratin in hair and nails, the filaments of the cytoskeleton that give a cell its shape. They transport molecules, like membrane channels that we will see in cell membranes or hemoglobin carrying oxygen. They move cells and organisms. The motor proteins in muscle fibers cause muscles to contract so we can walk and breathe. Proteins also act as signals. Many hormones are proteins or peptides like insulin, and as signal receivers, receptors on cell surfaces. They serve as antibodies of the immune system, defending us against pathogens. It's hard to find a process in life that does not involve proteins in some way. As you relax and absorb this, imagine proteins as diligent little entities, each folded into a particular shape, each doing a specific job quietly. Some are like scissors cutting molecules, others are like glue joining molecules, some are building scaffolds, and others carrying messages. All these proteins are composed of the same set of amino acid building blocks, yet each protein's unique sequence and shape determine its role. It's a beautiful demonstration of how complexity can arise from simple repeating units. Nucleic acids, the blueprint of life. Finally, we come to nucleic acids, the macromolecules that store and transmit genetic information. These are the famous DNA and RNA. A nucleic acid is a polymer made of monomers called nucleotides. Each nucleotide has three parts: a sugar, a phosphate group, and a nitrogenous base. The sugar in RNA is ribose, and in DNA is deoxyribose, which is like ribose but missing one oxygen atom. That is what the names stand for. DNA equals deoxyribonucleic acid, RNA equals ribonucleic acid. Attached to the sugar is a phosphate group, and linking through that phosphate from one sugar to the next is how nucleotides join in a chain, forming a sugar-phosphate backbone via phosphodiester bonds. The third component, the nitrogenous base, is the most interesting part because it carries the genetic code. There are 5 possible bases: adenine A, guanine G, cytosine C, thymine T, and uracil U. In DNA, the bases are A, G, C, and T. In RNA, A, G, C, and U. U replaces T. DNA is like a long molecular storybook that contains instructions for building and maintaining an organism. It usually exists as a double helix, a gently spiraling ladder-like structure. The two sides of the ladder are the sugar-phosphate backbones of 2 nucleotide chains, and the rungs of the ladder are pairs of bases, one from each strand, hydrogen bonded together. The pairing is very specific. A always pairs with T, and G always pairs with C in DNA. They fit together like 2 puzzle pieces, A with T via 2 hydrogen bonds, G with C via 3 hydrogen bonds. In RNA, which is usually single-stranded, you will pair with A if needed, like during transcription or in certain RNA secondary structures. But in DNA's double helix, this pairing rule ATGC means that the 2 strands are complementary to each other. If one strand has the sequence ACGT on one side, the opposite strand will be TGCA. The 2 strands also run in opposite directions. One seems upside down relative to the other in terms of orientation, something we call anti-parallel. This anti-parallel orientation and the base pairing allow DNA to be copied and read in a reliable way. The sequence of bases along a DNA strand encodes information, much like letters spell out sentences. A gene is a specific stretch of DNA that carries the instructions to make a particular protein, or sometimes an RNA molecule. We'll dig deeper into how those instructions are used when we touch on molecular genetics. For now, imagine the DNA in each of your cells as a long, coiled thread containing millions or even billions of base pairs, coiled up in chromosomes inside the nucleus of the cell. It's amazingly compacted. If you stretch the DNA from one human cell end to end, it would be about 2 meters long, yet it fits in a nucleus only a few micrometers wide. RNA is like DNA's working cousin. There are different forms of RNA in the cell, but the one many people know is messenger RNA, mRNA, which carries a copy of a gene's instructions out of the nucleus to the sites of protein synthesis. RNA is typically single-stranded and uses uracil in place of thymine. It can fold into interesting shapes too, and some RNAs have catalytic activity or other roles, like ribosomal RNA and transfer RNA, which we'll mention later. In summary, nucleic acids are the cell's information storage and retrieval molecules. DNA is the long-term archive, stable and passed from generation to generation, whereas RNA is more transient, often made as needed to carry instructions to ribosomes where proteins are assembled. We will soon explore how DNA is used to create RNA and then proteins, but for now, Just appreciate the gentle idea that in the nucleus of each of your cells, right now, there is a serene library of DNA with all the chapters of you written in a simple 4-letter chemical alphabet. The cell can read this information and execute its instructions methodically and calmly. We have now met the 4 major macromolecules: carbohydrates, lipids, proteins, and nucleic acids. Each plays vital roles. Carbohydrates fuel and form structures. Lipids store energy and build membranes, proteins do the work and provide structure, and nucleic acids carry the blueprint. These molecules interact beautifully. For example, DNA encodes proteins. Proteins, as enzymes, help build and break down carbohydrates and lipids. Lipids form membranes where proteins often reside, and so forth. Life is an interconnected web of these chemical pieces. Take a slow breath and consider how far we've come, from simple atoms joining in bonds, to water and simple compounds, and now to the macromolecules that make up living cells. It's a gentle yet incredible progression, a kind of assembly from simplicity to complexity. And yet, within the living cell, this assembly and transformation is happening constantly and effortlessly, guided by the laws of chemistry and the information in DNA. Before we proceed to look at whole cells, let us pause briefly to reinforce a key idea: enzymes. Enzymes are proteins, usually, that make the chemistry of life possible at mild temperatures and conditions. We've hinted at them, but now we will give them a closer, gentle look, because they truly deserve special attention for the role they play. Imagine them as quiet facilitators, enabling reactions to occur swiftly and smoothly. In our next section, we will explore enzymes and how they catalyze life's processes while maintaining our soft, calming narrative. Enzymes, nature's gentle catalysts. Even as you breathe softly and your heart beats steadily, inside you countless chemical reactions are occurring in each cell. Sugars are being broken down for energy, new molecules are being built up, old ones recycled. Normally, chemical reactions might be slow or require harsh conditions like high heat to proceed. But in the warm and mild environment of your body, reactions need a little help to go fast enough. This help comes from enzymes, which are biological catalysts. Enzymes are typically proteins, and a few are RNA molecules that speed up reactions without being consumed in the process. They provide a comfortable pathway for a reaction to occur, kind of like offering a smooth slide instead of a rocky climb. Every chemical reaction requires a certain amount of energy to get started. This is called the activation energy. You can imagine it as a small hill that reactant molecules must climb before they can roll down into products. Enzymes work by lowering that hill, making it easier for molecules to reach the top and slide down into a chemical reaction. They do this in various subtle ways, by bringing reactants together in the right orientation, by bending or straining bonds, by providing an environment that favors the reaction, or even by briefly participating in the reaction in a way that helps it along, only to return to their original state at the end. When an enzyme speeds up a reaction, we say it catalyzes the reaction. Each enzyme has an active site, a special pocket or groove in its folded protein structure where the reactant molecules, called substrates, fit like a key in a lock. The enzyme substrate fit is often extremely specific. An enzyme will typically act on only one type or a few types of substrate. When the substrate nestles into the active site, the enzyme might hug it slightly, changing shape in an induced fit that facilitates the reaction. It's a very gentle and precise mechanism. Picture the enzyme as a helpful guide that briefly holds the substrate in a perfect position or shape so that a chemical bond can break or form more easily. Without enzymes, the reactions in our bodies would happen far too slowly. For example, as mentioned, hydrogen peroxide, H2O2, decomposing into water and oxygen might take years to break down on its own. But with catalase enzyme present, it happens in milliseconds. Similarly, the digestion of food, the copying of DNA, the production of energy, all these processes rely on enzymes each step of the way. In fact, life as we know it is utterly dependent on enzymes to proceed at life-friendly temperatures. Enzymes are usually named after what they do, often ending in ase. A protease breaks down proteins, lipase breaks down lipids, DNA polymerase builds DNA polymers, ATP synthase synthesizes ATP, and so on. The names give hints of their function, but behind each name is a protein with a unique shape, facilitating a specific reaction in a calm, effective manner. Enzymes themselves function best under certain conditions, and each enzyme has its own preferred environment. Many human enzymes like a temperature around 37 degrees Celsius, our body temperature, and a neutral pH around 7, but some operate in acidic conditions like pepsin, which works in the stomach at pH 2. If conditions deviate too much, for example if the temperature gets too high, enzymes can denature, unfold, and lose activity. That's one reason high fevers are dangerous. The body's enzymes start to misbehave or stop working when heated too much. Likewise, extreme pH can protonate or deprotonate key amino acids in an enzyme's active site, disrupting its function. But under normal conditions, enzymes gently perform their tasks over and over, each enzyme molecule catalyzing reactions many times per second in some cases. Cells also regulate enzymes. They can produce fewer enzyme molecules as needed gene regulation, and they can also use inhibitors to slow enzymes down or activators to speed them up. There are molecules that can bind to an enzyme and block the active site. These are competitive inhibitors because they compete with the substrate for the same binding spot. Imagine a mimic that sits in the lock so the key substrate can't enter. Competitive inhibition can often be overcome by simply having more substrate. If there's lots of the real substrate, it will more likely win the competition and the enzyme will work. Then there are non-competitive inhibitors, which bind elsewhere on the enzyme and change its shape, so the substrate might still bind, but the enzyme won't catalyze effectively. This is like someone bending the lock, so even if the key can go in, it can't turn. Non-competitive inhibition cannot be overcome by adding more substrate, because the enzyme's function is essentially turned off by the shape change. Cells use such strategies to fine-tune metabolic pathways. In fact, often the end product of a pathway will inhibit an enzyme that acted earlier in the pathway, a form of feedback inhibition, ensuring that when enough product is made, the pipeline slows down. On the flip side, there are also allosteric activators, molecules that bind to a site on the enzyme, not the active site, and stabilize a shape of the enzyme that is active, thus enhancing its activity. It's as if a helper molecule locks the enzyme in a ready position so it works more efficiently. So enzymes are not just static workers, they are dynamic and regulated. This regulation keeps the cell's chemistry balanced. If you imagine again that cell city, enzymes are like efficient workers that can be scheduled or paused based on the needs of the city, ensuring no resources are wasted and everything runs smoothly. With enzymes assisting every reaction, the cell can maintain a gentle yet productive pace of life. Molecules break and form under the enzyme's guidance as softly as turning a page in a book. Swift, but gentle. You don't need to actively remember all these details. Just have the soft impression that enzymes are benevolent facilitators within you, making sure all the chemistry that needs to happen does happen, and at the right time and place. Now, having covered the molecular players, from small molecules to macromolecules and the enzymes that choreograph their reactions, it's time to step back and look at the cell as a whole. Up to now, we've been focusing on the ingredients and tools of life. Next, we will visit the cell, the basic unit of life, and see how these molecules come together to form a living, functioning unit. Continue to relax as we gently transition to exploring the cell, like entering a quiet and intricate little world of its own. The cell, a peaceful microscopic world. Let's now imagine ourselves drifting gently into a tiny, tiny world, the world of a single cell. The cell is often called the fundamental unit of life, like a small room where all the processes of life happen on a miniature scale. Some organisms, like bacteria or yeast, consist of just one cell. Larger organisms like ourselves have trillions of cells, each one alive and cooperating in the community that is our body. Regardless, each cell is a dynamic but orderly place, containing many parts that each have special roles, much like rooms and machinery in a house or factory. Picture a typical animal cell, soft and roundish, maybe like a little water balloon filled with semi-fluid cytoplasm. A plant cell is similar but often more rigid and rectangular because it has a sturdy cell wall outside its cell membrane. Both have many internal structures known as organelles, literally little organs. We will explore some key organelles now, moving gently from one to the next, as if strolling through a quiet little city inside the cell. Every cell is enveloped by a cell membrane, also called the plasma membrane. This membrane is like a thin, flexible skin that holds the cell together and controls what enters or leaves. The membrane is a phospholipid bilayer, as we talked about, with hydrophilic heads facing the watery outside and inside of the cell, and hydrophobic tails facing each other in the middle of the membrane. Embedded in this fluid mosaic of lipids are numerous proteins that serve various functions. Some act as channels or gates for molecules to pass through, others as receptors for signals, and others as anchors connecting to the cell's internal scaffolding or to neighboring cells. You can imagine the membrane as a calm sea of lipids with proteins floating within it, bobbing around. It's fluid and dynamic. Parts can move, come together or drift apart, yet it provides a reliable boundary. For plant cells, just outside the membrane is a cell wall made of cellulose, which is strong but porous. Think of it like a sturdy mesh or a framework that gives the cell shape and protection. Plant cell walls give plants their rigidity, which is why a celery stalk or a tree trunk can stand upright. Animal cells don't have cell walls. They rely on a more flexible structure and often connective tissues or external skeletons for support. Now, gently float inside the cell. The interior of the cell, the cytoplasm, is not empty. It's a crowded yet organized place. The cytoplasm is an aqueous environment rich in proteins, salts, and organelles. There is also a network of fibers called the cytoskeleton, which acts like an internal framework. It helps maintain the cell's shape, allows the cell to move if it's a crawling cell like an amoeba or a white blood cell, and provides tracks along which organelles and molecules can be transported like tiny cargo trains. Near the center of an animal cell, and typically off to the side in a plant cell due to a large vacuole, is the nucleus. This is like the cell's command center or library. The nucleus is a sphere or oval structure enclosed by a double membrane, the nuclear envelope, which has pores to allow materials in and out. Inside the nucleus is where the DNA resides, the chromosomes containing all the genetic information. The nucleus in our peaceful cell city is a quiet room where the instructions for life are stored and gently read. When a cell needs to make a protein, the corresponding gene's DNA in the nucleus is transcribed into messenger RNA, which then exits the nucleus through the nuclear pores to be translated into protein in the cytoplasm. There's also a region inside the nucleus called the nucleolus, which is like a little factory for assembling ribosomes, machines that will make proteins. Speaking of ribosomes, these are small, grain-like structures either floating in the cytoplasm or attached to certain membranes. Ribosomes are the sites of protein synthesis. They read the mRNA instructions and assemble amino acids into proteins. They are like tiny 3D printers, but instead of plastic, they print proteins. Many ribosomes stud the surface of a particular organelle called the rough endoplasmic reticulum, giving it a rough appearance under a microscope. Let's talk about that next. The endoplasmic reticulum, ER, is a network of membrane tubules and sacs that extends from the nuclear envelope throughout the cell. There are two types, rough ER, which has ribosomes on it, and smooth ER, which does not. The rough ER is like a conveyor belt for protein assembly, as ribosomes on its surface synthesize proteins, especially those that will be secreted from the cell or inserted into membranes. These proteins can be pushed into the interior of the ER to be folded and modified. The ER often adds carbohydrate side chains to proteins, forming glycoproteins, and helps them fold correctly. Think of rough ER as a series of hallways or channels where new proteins go for quality control and finishing touches, escorted by the walls, membranes themselves. Meanwhile, smooth ER lacks ribosomes and has different functions. Smooth ER is involved in lipid synthesis. It produces new phospholipids for the cell membrane, steroid hormones, and other lipids. It also helps in detoxification. For example, liver cells have a lot of smooth ER to help detoxify chemicals. And in muscle cells, a special kind of smooth ER stores calcium ions used for muscle contraction. From the ER, new proteins and lipids often are packaged into small membrane bubbles called vesicles and sent to the Golgi apparatus. The Golgi apparatus, or Golgi body, is like the cell's post office or shipping center. It looks like a stack of flattened pancakes, each a membrane-bound sac. Vesicles from the ER fuse with one side of the Golgi, delivering their cargo. Inside the Golgi, proteins and lipids might be further modified, perhaps more sugars added or phosphate groups attached and sorted. Then, new vesicles bud off from the other side of the Golgi, carrying these finished products to their destinations. Some will go to the cell membrane to secrete their contents outside. For instance, a cell in your pancreas might secrete insulin this way. Others will become lysosomes or other organelles. The Golgi ensures everything is properly addressed and packaged before it leaves. One type of vesicle that buds off the Golgi is the lysosome in animal cells. Lysosomes are like little recycling centers or digesters. They contain enzymes that can break down biomolecules, proteins, lipids, carbohydrates, nucleic acids into their monomers. If a cell engulfs a food particle or a bacterium, that gets delivered to lysosomes for digestion. Lysosomes also digest worn-out organelles so their components can be reused. They work best at an acidic pH, which is maintained inside the lysosome like a contained compost bin. In our calm cell narrative, lysosomes quietly keep things tidy, breaking down wastes and old parts so the building blocks can be repurposed. Plant cells don't have lysosomes. Instead, they have one or more large vacuoles that perform similar functions and more. A central vacuole in a plant cell can occupy a big portion of the cell's volume. It stores water, nutrients, pigments, and waste products. By accumulating water, the vacuole helps maintain turgor pressure, the pressure of the cell contents against the cell wall, which keeps the plant upright and the leaves perky. In flowering plants, vacuoles might contain colourful pigments like the red in rose petals, or defensive poisons to deter herbivores. In our mind's eye, a plant cell's vacuole is like a large serene pond in the middle of a walled garden, storing vital liquids and solutes, keeping the cell plump and supported. Now, a very important organelle, the mitochondrion, plural mitochondria. These are often dubbed the powerhouses of the cell because they are the site of cellular respiration, the process of generating ATP, the cell's energy currency from food molecules. Mitochondria are small ovoid or sausage-shaped structures with a double membrane. The inner membrane is highly folded into cristae, giving it a large surface area. These folds are studded with the machinery for producing ATP. The outer membrane encloses everything like a smooth cover. Inside the inner membrane is the mitochondrial matrix, which contains enzymes, mitochondrial DNA, and ribosomes. Interestingly, mitochondria are thought to have originated from free-living bacteria that took up residence inside early eukaryotic cells, forming a symbiotic relationship. This is the endosymbiotic theory. That's why mitochondria have their own small circular DNA and replicate somewhat independently. In our cell's quiet city, the mitochondria are like efficient power plants humming along, using oxygen and nutrients, like glucose, to produce ATP energy molecules. We will explore how exactly they do that when we discuss respiration, but for now just know that when you inhale and exhale, you are supplying oxygen to these mitochondria and removing the carbon dioxide they produce, keeping the energy production going. In plant cells and some algae, there's another energy-related organelle, the chloroplast. Chloroplasts are the sites of photosynthesis, where light energy is captured and used to build sugars from carbon dioxide and water. They are typically green because of the pigment chlorophyll. Like mitochondria, chloroplasts have a double membrane and their own DNA, and they are also believed to have originated from symbiotic bacteria, in this case, cyanobacteria. Inside chloroplasts are stacks of membrane sacs called thylakoids, where the light reactions occur, and the surrounding fluid called stroma, where the Calvin cycle, the sugar-building phase, occurs. We will talk about photosynthesis soon in more detail. For now, if our cell was a plant cell, we'd see chloroplasts as little green energy factories that work in daylight, quietly producing food, glucose, and releasing oxygen. At night, or in non-photosynthetic cells, mitochondria take over in using that food for energy. Let's not forget some additional players in cells. Centrioles in animal cells help in cell division. They organize microtubules that pull chromosomes apart during mitosis. Peroxisomes help break down certain chemicals like fatty acids and toxins with oxidative reactions, producing hydrogen peroxide, which they then decompose safely. And in multicellular organisms, cells often have specialized structures. For example, cells lining our respiratory tract, have tiny hair-like cilia that beat to move mucus. Sperm cells have a flagellum, a long tail to swim. Some cells have microvilli to increase surface area for absorption. The cell truly is like a tiny, busy world. But even in its busyness, in the context of our slow, meditative narration, you can imagine each organelle and component doing its job calmly and reliably. There's a rhythm. The nucleus issues instructions. Ribosomes build proteins. The ER and Golgi fold and ship them. Mitochondria generate energy. Lysosomes clean up. It all happens in harmony, maintaining the cell's life. One thing to appreciate is that cells maintain a balance of internal conditions. They keep a stable environment inside despite changes outside. This steady state is part of what's called homeostasis, which we'll explore soon on a larger scale. Inside the cell, the membrane's selective permeability helps maintain the right ion concentrations, pH, and nutrient levels. The nucleus controls which proteins to make, adjusting to the cell's needs. If the cell is stressed or lacking something, it has ways to respond, like sending signals to get more nutrients or to slow down growth. Yet, to a large extent, a cell is self-regulating, a tiny autopilot ensuring everything stays just so. Before we move on from the cell, imagine yourself gently floating out of that cell city and seeing others like it around. In a multicellular organism like us, cells exist in communities, forming tissues. They stick to each other via cell junctions and communicate through chemical signals or direct contact. Some cells specialize: nerve cells, muscle cells, blood cells, etc., each with the same basic organelles but tweaked structures and functions to serve a role in the organism. It's as if each cell type is a different character in a story, yet all of them share the same fundamental cellular machinery we described. Now that we have an overview of the cell's structure and components, we are ready to look at some specific processes that happen in cells. Two of the most crucial processes are how cells get energy from food and how plants capture energy from light. These are cellular respiration and photosynthesis. Beautifully complementary processes. They are a bit more chemistry heavy, but we will approach them in a light and easy manner, focusing on the big picture and maintaining our slow and gentle pace. Take another deep breath, feel free to adjust your position for comfort, and let's continue our journey into the realm of metabolism and energy flow in life.
Metabolism.
The cell's gentle energy conversions. Every living cell is like a tiny engine, constantly running many chemical reactions to sustain life. The sum total of all these chemical reactions is known as metabolism. Metabolism encompasses both building things up and breaking things down, all quietly orchestrated by enzymes. When the cell builds larger molecules from smaller ones, for example, when it links amino acids to form a protein or joins sugars to form starch, this is called anabolism or anabolic reactions. Anabolic reactions usually consume energy. They are like construction projects that require an input of fuel or ATP to proceed. On the other hand, when the cell breaks down larger molecules into smaller pieces, like digesting glucose for energy or breaking down fats, this is called catabolism or catabolic reactions. Catabolic reactions typically release energy, often capturing some of it in the form of ATP. A simple way to remember: anabolic builds, think add; catabolic cuts, think catastrophe or breakdown. Together, anabolism and catabolism are the two sides of metabolism, and they are balanced and coordinated within the cell. Let's consider how energy works in these reactions. In chemical terms, energy is the capacity to do work or cause change. In cells, energy often comes in the form of chemical potential energy stored in bonds. A molecule of glucose, for instance, contains energy in the bonds holding its atoms together. When those bonds are rearranged or broken in a catabolic reaction, energy can be released. But the cell can't directly use the energy of glucose all at once. Doing so would be like a sudden combustion, too uncontrolled and not directly useful for powering cellular activities. Instead, the cell uses a kind of energy currency, ATP, adenosine triphosphate. Think of ATP as a small battery or a charged spring. It consists of adenosine, which is adenine plus a sugar, attached to 3 phosphate groups. Those phosphate groups are linked by high-energy bonds. When a cell needs energy, it can break the bond of the last phosphate, converting ATP to ADP, adenosine diphosphate, and a free phosphate, and in doing so release energy that can drive a reaction or do work. This could be used for muscle contraction, for transporting substances across membranes, for building molecules, and so on. Then the cell can recharge AP back into ATP by adding a phosphate again, but that requires energy input, which usually comes from food breakdown. So ATP is continuously cycled, charged up with energy from catabolism, then used to power anabolism or other work, then charged up again. It's much like a rechargeable battery within the cell, ensuring energy is readily available in small, manageable packets. Now, how does the cell charge up ATP? This is where the core processes of cellular respiration and, in plants, photosynthesis come in. These processes are about energy transformation. Cellular respiration is how cells extract energy from food, like glucose, in the presence of oxygen and store it in ATP molecules. Photosynthesis is how plants and some bacteria capture energy from sunlight and store it in glucose, which can later be used in respiration. It's helpful to think of these processes as mirror images in a way. In cellular respiration, glucose, C8H2O and oxygen-O2 are used up, and carbon dioxide, CO2, water, H2O, and energy, ATP, are produced. In photosynthesis, carbon dioxide and water are used, and glucose and oxygen are produced. It's a beautiful cycle. Animals and other non-photosynthetic organisms breathe in oxygen and eat organic carbon, like plant sugars or other organisms, and breathe out carbon dioxide and water, capturing energy for life. Plants take in that carbon dioxide and water, use sunlight to make sugars, food, and release oxygen. It's as if animals and plants are engaged in a slow planet-wide dance of gas exchange and energy exchange, supporting each other. We'll discuss respiration first, since it happens in almost all cells, plant or animal, and then photosynthesis. Cellular respiration, energy from food. Cellular respiration is the process by which cells generate ATP by breaking down glucose or other organic fuels with the aid of oxygen. It's like a very controlled burning of sugar. If you were to burn sugar in a flame, you'd get carbon dioxide, water, and heat in one big burst. Cells do essentially the same chemistry, but in dozens of smaller enzyme-guided steps, capturing much of that energy in ATP rather than letting it all escape as heat. This way, the cell harnesses the energy for useful work. The overall simplified equation for aerobic cellular respiration is glucose plus 6O2, 6CO2 plus 6H2O plus energy ATP. This means one molecule of glucose combined with 6 molecules of oxygen yields 6 molecules of carbon dioxide and 6 molecules of water, and in the process energy is released and trapped in ATP. In reality, about 40% of the energy in glucose is captured in ATP, and the rest is released as heat, which in warm-blooded animals helps maintain body temperature. Cellular respiration consists of 4 main stages, which we can walk through in a gentle overview: glycolysis, pyruvate oxidation, the Krebs cycle, also called the citric acid cycle, and the electron transport chain and oxidative phosphorylation. 1. Glycolysis. This word literally means splitting sugar. It happens in the cytoplasm of the cell and does not require oxygen directly, it's anaerobic. In glycolysis, one molecule of glucose, 6 carbons, is split into 2 molecules of pyruvate, each 3 carbons. Along the way, a little energy is released, enough to produce a net gain of 2 ATP molecules. Actually, 4 ATP are produced, but 2 were used to kickstart the process, so the net is 2. Also, some high-energy electrons are captured in carriers called NADH. NADB picks up electrons and H to become NADH. Think of NADH as a charged shuttle that will carry electrons to the last stage. Glycolysis is like the initial breakdown of glucose into smaller pieces and yields a small energy payoff, ATP. And some electron carriers, NADH. It's relatively quick and doesn't yield much ATP, but sets the stage for further breakdown. 2. Pyruvate oxidation. After glycolysis, the 2 pyruvate molecules, each 3 carbons, are transported into the mitochondria. There, each pyruvate is converted into a molecule called acetyl-CoA, 2 carbons attached to a coenzyme A. In this conversion, 1 carbon from pyruvate is released as CO2, so 2 CO2 per glucose since there were 2 pyruvates, and more electrons are transferred to NAD to form NADH. No ATP is made in this step directly. Its main purpose is to produce acetyl-CoA, which can enter the next cycle, and to yield a bit more NADH. So now, glucose's carbon skeleton has been reduced from 6 carbons, glucose, to 2 fragments of 2 carbons, the acetyl groups, with 2 carbons already gone as CO2. Those acetyl groups on CoA carry potential energy into the Krebs cycle. Number 3, Krebs cycle, citric acid cycle. Acetyl-CoA enters this cycle in the mitochondrial matrix. It's a cyclic series of reactions that finishes the oxidation of the organic fuel. Each 2-carbon acetyl group is attached to a 4-carbon molecule at the start of the cycle, then progressively broken down, releasing 2 CO2 molecules, regenerating the 4-carbon starting molecule for the next turn. For each acetyl-CoA, the cycle produces 1 ATP, but more significantly, multiple electron carriers, 3 NADH and 1 FADH2, a cousin of NADH. Since 2 acetyl-CoA came from 1 glucose, the cycle runs twice per glucose, yielding 2 ATP, 1 each cycle, 6 NADH, 2 FADH2 in total, and releasing 4 CO2 combined from both cycles. By now, all 6 carbons of the original glucose have been released as CO2, 2 in pyruvate oxidation, 4 in Krebs cycle. What remains are the electrons gathered by NADH and FADH2 and a couple of ATP. The Krebs cycle is like a gentle wheel that picks up the acetyl group, processes it, and spits out carbon dioxide, turning the handles to load up high-energy electrons into NADH and FADH2. The direct ATP gain is small, but the real value is in those loaded electron carriers, which hold a lot of potential energy. Number 4, electron transport chain ETC and oxidative phosphorylation. This is the final stage and where the big ATP payout happens in the inner mitochondrial membrane. All the NADH and FADH2 molecules from prior steps come here to deliver the electrons they carry. They basically say, here, take these electrons and use them. The electron transport chain is a series of protein complexes and other molecules embedded in the inner membrane. Electrons from NADH and FADH2 are passed along this chain from one carrier to the next in a series of redox reactions. As electrons flow through the chain, they release energy bit by bit. This energy is used to pump protons H-pine from the mitochondrial matrix into the intermembrane space, the area between the inner and outer membranes. It's like using the energy of electrons to wind up a spring or pump water uphill. Here, the spring, or water, is a proton gradient, a higher concentration of protons outside the inner membrane than inside. This gradient stores energy, like a dam holding water. At the end of the chain, electrons have lost much of their energy. They need to go somewhere, and here is where oxygen comes in. Oxygen is the final electron acceptor. Each oxygen atom accepts electrons and combines with protons to form water. This is why we need oxygen, to clean up those electrons and protons and form water. If oxygen isn't present, the whole chain backs up and stops, which is why without oxygen, anaerobic conditions, the ETC can't run and the Krebs cycle can't run either, because NADH has nowhere to unload its electrons. That's when cells resort to fermentation, like lactic acid fermentation in mussels or ethanol fermentation in yeast, to regenerate some NADD, but those yield far less ATP. But in the comfortable presence of oxygen, the chain flows smoothly. Now we have a proton gradient established outside the inner membrane, like a reservoir of potential energy. The protons want to flow back into the matrix because of both concentration and charge differences. They flow in through a special enzyme called ATP synthase, which is also embedded in the inner membrane. Think of ATP synthase as a tiny turbine or water wheel. As protons rush through it, it spins and uses the energy of that flow to synthesize ATP from ADP and phosphate. It's a brilliant molecular machine. This process of using a proton gradient to drive ATP synthesis is called oxidative phosphorylation because it's powered by the redox reactions of electron transport with oxygen involved. It's also an example of chemiosmosis, using a chemical gradient to do work. Osmosis of protons, essentially. How much ATP is made? From one glucose, the entire process of respiration can yield around 30 to 38 ATP molecules, depending on the cell conditions and efficiencies. Often you see the number 36 or so in textbooks for eukaryotic cells. This is vastly more than the 2 ATP from glycolysis alone. So the electron transport and ATP synthase step is where the majority of energy payoff comes. It's the difference between sprinting a few steps and running a marathon, energy-wise. In summary, cellular respiration takes the energy from glucose and, through a series of gentle, enzyme-guided steps, deposits it into ATP. Carbon dioxide and water are left as byproducts, which our bodies simply exhale and excrete without a second thought. But those CO2 molecules we breathe out were each part of a sugar molecule that was broken down, and their breakdown helped recharge our ATP batteries. Let's take a calming moment to visualize the whole respiration process in a soft way. Imagine eating a grape. It contains sugars that were made by a plant from sunlight. As you relax, your body digests that grape, and the glucose enters your cells. In a muscle cell, that glucose is gradually taken apart. In the cytoplasm, it's split in two, glycolysis. Then in the mitochondria, the fragments are further broken down and their electrons collected. Your mitochondria, like tiny power stations, use oxygen you breathed in to accept those electrons, and in doing so, pump protons and create a flow that spins ATP synthase. With each quiet turn of this molecular turbine, an ATP is made, ready to supply energy when that muscle cell decides to contract or do any work. It's all happening quietly, without any conscious effort, like a well-tuned engine idling smoothly. You only become aware of it, perhaps when you exercise vigorously and start breathing heavier. That's your body delivering more oxygen to ramp up those mitochondria to produce more ATP for hard-working muscles. But at rest, it's tranquil. Your cells respire, your heart and lungs provide the O2 and remove the CO2, and you remain comfortably unaware, free to drift into sleep if you wish. Knowing how respiration works, we can appreciate why we breathe and why we eat in simple terms. Breathing brings in oxygen for the ETC and removes the CO2 from Krebs cycle. Eating provides the glucose and other fuels that feed into glycolysis and Krebs. It's a partnership between our cells and the environment. The air and food we take in get integrated into this cycle of energy transformation. Now, as promised, we will look at the complementary process, photosynthesis, which is essentially where the glucose originally comes from, in an ecological sense. While respiration occurs in almost all life forms, even plants respire, especially at night or in their non-green tissues, photosynthesis occurs in plants, algae, and some bacteria. It captures energy from the sun and stores it in chemical bonds of sugar. This in turn provides food, either directly or indirectly, for all other organisms. It's a beautiful process that fills our world with energy and oxygen, and it does so in a calm, green, and growthful way. Photosynthesis, energy from light. Now let's gently turn our attention to the green leaves of plants, or perhaps to algae floating in a quiet pond, or even to the tiny cyanobacteria in the oceans. These organisms perform photosynthesis, a process that is, in a way, the reverse of respiration. If respiration is like a controlled burning of sugar to harvest energy, photosynthesis is like a quiet capturing of solar energy to build sugar. It's as if plants have little solar panels in their cells, the chloroplasts, where they take sunlight, water, and carbon dioxide and convert them into chemical energy stored in glucose, all while releasing oxygen as a byproduct. The simplified overall equation for photosynthesis is essentially the opposite of respiration's. 6 CO2 plus 6 H2O plus light energy, C-Sieve H2O-Sieve, glucose plus 6 O2. This means 6 molecules of carbon dioxide from the air and 6 molecules of water are combined using the energy of sunlight to produce 1 molecule of glucose and 6 molecules of oxygen, which get released to the atmosphere. This doesn't happen in one step, of course. Photosynthesis has 2 main sets of reactions: the light-dependent reactions and the light-independent reactions. The latter are also known as the Calvin cycle or dark reactions. Let's walk through it calmly. 1. Light-dependent reactions. Photostage. These occur in the thylakoid membranes inside the chloroplast. Thylakoids are like flattened sacs or discs arranged in stacks grana. Embedded in their membranes are pigment molecules, primarily chlorophyll, which is the pigment that gives plants their green color. Chlorophyll and other accessory pigments absorb light energy, mainly red and blue wavelengths, reflecting green, thus we see leaves as green. When chlorophyll absorbs a photon of light, one of its electrons gets excited to a higher energy level. This excited state doesn't last long. The chlorophyll passes the excited electron through a series of carriers, similar in spirit to the ETC in respiration. In fact, plants have 2 main photosystems, 2 and I, named in the order of discovery, not sequence of action, that work together to capture light and produce energy-rich molecules. In photosystem 2, the energy from light is used to split water molecules into oxygen, protons, and electrons. This is called photolysis of water. Oxygen is released. All the oxygen we breathe comes from this splitting of water in photosynthesis, and the electrons from water replace those lost by chlorophyll. As the excited electrons travel through an electron transport chain in the thylakoid membrane, they release energy which is used to pump protons into the thylakoid interior, creating a proton gradient, much like in mitochondria. This gradient then drives ATP synthase to produce ATP, but now it's happening in a chloroplast. This process of making ATP using light energy is called photophosphorylation, and since electrons flow linearly from water to NAD+, forming NADPH, which we'll mention in a moment, it's often the non-cyclic electron flow. After giving up energy to make ATP, the electrons eventually reach photosystem 1, where they get a second boost of light energy. Another photon excites them again. These re-energized electrons then are passed to NADP, nicotinamide adenine dinucleotide phosphate, a cousin of NAD, along with a proton, reducing it to NADPH. NADPH, like NADH, is an electron carrier. It holds high-energy electrons and a proton. So the products of the light-dependent reactions are ATP, energy currency, and NADPH, reducing power, and of course, O2 as a byproduct from the water splitting. We can think of ATP and NADPH as the usable forms of solar energy. They carry the captured sunlight energy in chemical form. These products will be used in the next stage, the Calvin cycle, to build glucose. There's also a possibility of cyclic electron flow, where electrons from photosystem 1 loop back to the earlier part of the chain to make extra ATP without producing NADPH. Plants do this when the ATP demand in the Calvin cycle is higher relative to NADPH. 2. Light-independent reactions, Calvin cycle. These reactions don't require light directly. They can happen in light or dark as long as ATP and NADPH are available, and occur in the stroma of the chloroplast, the fluid outside thylakoids. The purpose of the Calvin cycle is to take CO2 from the air and use the energy of ATP and NADPH from the light reactions to synthesize glucose, or more accurately, a 3-carbon sugar that can be later combined to form glucose. The Calvin cycle is sometimes summarized in 3 phases: carbon fixation, reduction, and regeneration. In the first phase, carbon fixation, the enzyme Rubisco, one of the most abundant enzymes on Earth, attaches CO2 to a 5-carbon sugar called RuBP, ribulose bisphosphate. This forms an unstable 6-carbon compound that immediately splits into 2 3-carbon molecules, 3-phosphoglycerate. Rubisco is a bit slow and plants need a lot of it, but it works steadily, drawing in CO2 and fixing it into organic form. For every CO2 fixed, 2 molecules of 3-carbon product are made. Since the cycle will run for 3 CO2 molecules at a time, for reasons of stoichiometry with 5-carbon RuBP, you get 6 3-carbon molecules at this point. Next, in the reduction phase, ATP and NADPH from the light reactions are used to convert those 3-carbon molecules, 3-PGA, into a higher energy form called G3P, glyceraldehyde 3-phosphate. Specifically, ATP phosphorylates the 3-PGA, adding a phosphate, and NADPH donates electrons, reducing it to create G3P. G3P is a 3-carbon sugar, one that can be considered a building block for glucose. Indeed, after 3 CO2 have entered, one cycle encompassing 3 CO2, 6 G3P molecules are produced. One of those 6 is output from the cycle. This is the net gain, which can go on to help form glucose or other carbohydrates, and the other 5 G3Ps are used in the next phase to regenerate the starting RuBP. The last phase is regeneration of RuBP. It's a bit of a complex shuffle. 5 G3P, 3 carbon each, totaling 15 carbons, are rearranged using some more ATP into 3 molecules of 5-carbon RuBP, again 15 carbons total. This allows the cycle to continue, ready to fix more CO2. In summary, for every 3 CO2 molecules fixed, 1 G3P is netted. To get 1 glucose, which is 6 carbons, you'd need 2 G3P molecules. That typically means running the cycle 6 times, fixing CO2 to net 2 G3P, which can form glucose. It's intricate, but the gist is, using CO2 plus ATP and NADPH from the light stage, the plant assembles an energy-rich sugar, G3P, from which glucose fructose can be made by further processes. The ATP provides energy, and NADPH provides the reducing power, electrons. to turn CO2, which is low energy, oxidized carbon, into sugar, which is high energy, reduced carbon. The whole Calvin cycle is like a quiet factory in the stroma. CO2 comes in through stomata, tiny pores under leaves, diffuses into chloroplasts, ATP and NADP enter from the thylakoids, and out comes G3P, which the plant cell can use to build glucose, sucrose, starch, cellulose, whatever it needs. While all this is happening, we might envision a leaf basking in sunlight. The light reactions in the chloroplasts of that leaf produce oxygen as a waste product, which leaves through the same stomata. Meanwhile, water is being pulled up from the roots to supply the photosystems and also to keep the plant hydrated. Often in the day, plants make more oxygen and sugar than they immediately need, so they release the oxygen into the air, for us thankfully, and store the extra sugar as starch or other carbohydrates. At night, plants can break down that starch a bit to continue their metabolism, and they also respire like we do, using oxygen and giving off CO2, but usually they produce more O2 in the day than they consume at night, so net they provide oxygen. A fascinating aspect, photosynthesis is not 100% efficient either. It's influenced by factors like light intensity, CO2 concentration, temperature. If it's very bright, a plant can only go so fast because it might run out of CO2 or the Calvin cycle saturates. If CO2 is low, photosynthesis slows. If it's too hot or too cold, the enzymes like Rubisco don't work optimally. Plants have adaptations. For example, on very hot days, they may close stomata to prevent water loss, but that also means CO2 entry stops and O2 builds up. This can cause photorespiration, where Rubisco grabs O2 instead of CO2, leading to a wasteful process. Some plants, called seafraw or cam plants, have evolved ways to minimize photorespiration and conserve water, like cacti, which take in CO2 at night. But those details go deep. At a high level, it's fine to remember that most plants just do the Calvin cycle, C3 photosynthesis, and manage okay unless under extreme conditions. In our calm journey, let's imagine sitting under a tree on a sunny day. The leaves above are quietly at work. Sunlight is streaming down, chlorophyll is absorbing it, water from the soil is being split to give fresh oxygen into the air, and sugars are being produced that later might become part of an apple, or a grain of rice, or a potato underground. There is something profoundly peaceful about photosynthesis. It is life gathering energy in a gentle way, with sunlight and air, giving out oxygen in return. It happens in quiet green sanctuaries across the world, fueling ecosystems. With photosynthesis, we complete the loop. Plants make O2 and glucose. Animals and plants themselves at night use O2 to break down glucose, releasing CO2 and H2O, which plants use again. The sun's energy thus flows into living systems and is handed off in chemical form through food webs. Let's take a moment to appreciate this balance. In the day's light, there is a gracious excess of oxygen produced by photosynthesizers. This oxygen fills our atmosphere, a gift from the green world, which we and other aerobic organisms breathe. At night, or deep in the ocean where light doesn't reach, organisms continue to respire, but the oxygen is not depleted overall because of the constant renewal by plants and algae elsewhere. It's a beautiful equilibrium that has kept our atmosphere and life stable for millions of years. Now, having delved into biochemistry, cells, and core energy processes, we shift our focus from the microscopic back toward the larger scale of organisms. The energy from cellular respiration powers not just the cell's internal work, but also everything an organism does: movement, growth, thinking, maintaining body heat, etc. Likewise, the sugar produced by photosynthesis feeds not just the plant's cells, but animals that eat the plant, and so on. From here, we will explore molecular genetics, how the information in DNA guides the construction of those proteins and ultimately orchestrates life. Then we'll flow into how organisms maintain internal stability, homeostasis, and coordinate their functions with the help of the endocrine and nervous systems. We'll do all this continuing our gentle tone, ensuring the transitions are soft. Take a slow, deep breath if you like, and feel how the oxygen nourishing your cells might have been on a leaf moments ago. Feel gratitude for that exchange and ease as we proceed to uncover the secrets of the genetic code in a calm and accessible way. Molecular genetics, the whisper of the DNA. Deep inside each cell, in the quiet sanctuary of the nucleus for eukaryotic cells, or in the cell's cytoplasm for simpler bacteria, lies the genetic material, DNA, deoxyribonucleic acid. This long molecule holds the instructions for life, written in a chemical code. In our calm exploration, we will now look at how DNA stores information and how that information is used to build proteins, essentially guiding the cell's activities. We will also gently touch on how DNA is copied and how traits are passed on, keeping things straightforward and soothing. DNA structure and storage. DNA is famously structured as a double helix, which looks like a twisted ladder. The sides of the ladder are made of alternating sugar, deoxyribose, and phosphate units. This is the sugar-phosphate backbone of each strand. The rungs of the ladder are pairs of nitrogenous bases, adenine A, thymine T, cytosine C, and guanine G. A always pairs with T, and C always pairs with G via hydrogen bonds forming the rungs. This complementary base pairing is critical. It means if one strand has the sequence ATCG on one side, the opposite strand will have TAGC, perfectly complementary. Each strand of DNA is like a mirror template of the other, just with A swapped for T and G for C. The strands run in opposite directions, anti-parallel, but we need not worry about that detail too much, just that they are aligned in opposite orientation, which is important for copying and reading the code. The sequence of bases along the DNA, like ACG to CGA, is the code that determines everything about an organism, in conjunction with environment, of course. It's analogous to letters in a sentence. Groups of bases will encode specific amino acids in proteins, and genes are essentially sentences, or sometimes long paragraphs, that tell the cell how to make a protein. In human cells and other complex organisms, the DNA double helix is organized into structures called chromosomes. Humans have 46 chromosomes in most cells, 23 pairs, with each chromosome containing a very long DNA molecule and associated proteins, histones. If you stretched one cell's DNA out, it would be about 2 meters long, but through clever packing, wrapping around histones like thread around spools, then coiling further, it fits into a tiny nucleus. During most of the cell's life, the DNA is in a relaxed state, chromatin, accessible for use. Only during cell division does it condense into the classic X-shaped chromosomes we often see in images. Before a cell divides, it must replicate its DNA so that each new cell gets a complete set. DNA replication is a carefully orchestrated process. It begins at specific sites, origins, where the 2 strands of DNA are separated. Enzymes called helicases unwind and unzip the double helix, breaking the hydrogen bonds between bases to create 2 single-stranded templates. Each original strand serves as a template for a new strand, thanks to base pairing rules. Another enzyme, DNA polymerase, then comes in and adds new nucleotides one by one, complementary to the template strand, A to T, T to A, C to G, G to C. It builds the new strand in a specific direction, 5' to 3' direction if we get technical. So one strand, the leading strand, can be built continuously, while the other, the lagging strand, is built in short segments, Okazaki fragments, that are lay-joined by DNA ligase. It's as if one side of the ladder is copied smoothly and the other side is copied in pieces that are stitched together. Various proofreading mechanisms are in place. DNA polymerase itself can correct some mistakes. It has a proofreading function, and other enzymes can cut out mismatched bits, nucleases, and replace them. This ensures the copy is extremely accurate. Errors happen maybe once in a billion bases, and even those are often corrected. It's important to have fidelity because these instructions are vital. So each cell division, when your body's cells replace themselves or when an embryo's cells multiply, involves replicating all that DNA. In a meditative sense, one can imagine the double helix gently unzipping like a fermata in music, then 2 new complementary strands forming alongside the old ones, resulting in 2 identical double helices, each half old, half new. This is indeed called semi-conservative replication, since each new DNA helix conserves 1 old strand and has 1 new strand. After replication, when the cell divides, each daughter cell gets one of these identical DNA copies. From DNA to protein, gene expression, having DNA is like having a library of recipes. But how do those recipes get used to make actual dishes, proteins? The process of going from DNA to protein involves 2 major steps: transcription and translation. Transcription is the process of copying a segment of DNA, a gene, into RNA. Specifically, it makes messenger RNA, mRNA, which carries the information out of the nucleus to where proteins are made. Think of transcription as when you have a reference book in a library, which you can't remove from the library, so you copy the needed passage onto a piece of paper to take home. Similarly, DNA remains in the nucleus for eukaryotes, but an RNA copy of the needed gene is made so it can travel to the cytoplasm. During transcription, an enzyme called RNA polymerase binds to the gene's start, a sequence called the promoter. It prees the DNA strands apart just in that area and reads one of the strands, the template strand. It synthesizes a single-stranded RNA molecule by adding RNA nucleotides that are complementary to the DNA template, except RNA uses uracil, U, instead of thymine, T. For example, if the DNA template has a sequence ATCG, the RNA polymerase will add UAGC to the growing RNA. The RNA chain elongates until RNA polymerase reaches a termination signal in the DNA, at which point it stops and releases the RNA. So now an mRNA is produced, which has a sequence corresponding to the gene's coding sequence, similar to the non-template DNA strand, but with U in place of T. In eukaryotes, this initial mRNA is like a rough draft called pre-mRNA. It often contains extra bits, introns, that are not needed for protein coding, and these are removed in a process called RNA splicing, while the remaining cis exons are stitched together. Additionally, a protective cap and tail are added, 5' cap and poly-A tail. Once processed, the mRNA leaves the nucleus through a nuclear pore and enters the cytoplasm. Now comes translation, the process of reading the mRNA to build a protein. This occurs on ribosomes, which can be free in the cytoplasm or on the rough ER. Ribosomes are like little protein-making factories, made themselves of protein and ribosomal RNA, rRNA. The mRNA carries information in a code of 3-letter words called codons, where each codon corresponds to one amino acid, or a start-stop signal. For instance, the codon AUG means the amino acid methionine and also serves as a start signal. UUU means phenylalanine, and so on. This code is universal in life's domains, which is quite poetic in its consistency. Transfer RNA, tRNA molecules act as interpreters during translation. Each tRNA carries a specific amino acid and has a 3-nucleotide region called an anticodon that is complementary to an mRNA codon. For example, if a codon on mRNA is GGC, a tRNA with anticodon CCG will bind to it, and that tRNA will be carrying the amino acid glycine, since GGC codes for glycine. Translation goes like this. A ribosome assembles at the start codon of the mRNA, usually AUG. A tRNA with the complementary anticodon UAC, carrying methionine, comes in and binds. This sets the reading frame. Then the ribosome moves to the next codon, and the appropriate tRNA comes in with the next amino acid. The ribosome links the amino acids together with a peptide bond. Then it moves on. The first tRNA leaves without its amino acid, which is now joined in the chain, and the process repeats codon by codon, adding one amino acid at a time to the growing polypeptide chain. It's a remarkably coordinated process, kind of like reading a tape and assembling a product sequentially. When the ribosome hits a stop codon like UAA or UGA, which don't code for any amino acid, the process stops. The completed polypeptide is released and the ribosome disassembles. The new protein then folds up into its functional shape, maybe with help from chaperone proteins, and goes off to do its job, whether it's acting as an enzyme, a structural fiber, a hormone, etc. So, in summary, DNA, RNA, protein is the flow of genetic information, often termed the central dogma of molecular biology. DNA transcription produces mRNA, mRNA translation produces a protein. The whole process is called gene expression, turning the information in a gene into a tangible product, a protein, that affects the cell's structure or function. Only a fraction of a cell's genes are expressed at any one time. Cells have elaborate controls to turn genes on or off as needed, gene regulation. That's why a muscle cell and a nerve cell, which have the same DNA, look and act differently. They express different sets of genes. We can think of gene expression in a soothing way. Inside each nucleus, there's a library of blueprints, DNA. When a cell needs a particular protein, it gently finds the corresponding gene, makes an RNA copy, which then go to the busy workshop of the cytoplasm, where ribosomes quietly and diligently assemble the protein based on that copy. It's like ordering a specific part from a catalogue, then the workshop reads the design and builds the part. The role of DNA in heredity. We should touch on how DNA is the hereditary material, meaning it's from parents to offspring. When organisms reproduce, they pass on DNA, or in the case of some viruses, RNA to the next generation. In sexual reproduction, DNA from both parents combines to form a new unique combination, half from mother, half from father in humans, for example. In asexual reproduction, like bacteria dividing or a plant cutting growing into a new plant, the offspring's DNA is a copy of the single parent's DNA, barring any mutations. Mutations are changes in the DNA sequence. They can happen due to errors in replication not corrected, or due to environmental damage like UV light causing a base to change. Most mutations either have no effect or are harmful, but occasionally they can confer a benefit or new trait. Mutations in reproductive cells can be passed to offspring and are the raw material for evolution over long timescales. The diversity of life has arisen because of changes in DNA over eons, selected by the environment. It's also worth noting some tools of molecular genetics that humans have developed, which we will mention more in the next section on genetic tools, such as how we can intentionally cut and splice DNA using restriction enzymes. Amplify DNA with PCR, polymerase chain reaction, or sequence DNA to read the base code. These come from understanding how DNA works naturally and then harnessing those processes. But before diving into biotech, let's consider the entire journey of information. The quiet DNA holds codes that, when the time is right, are transcribed to RNA and translated to proteins. Those proteins may become enzymes that, for example, govern metabolism, or structural proteins that maintain cell shape, or signals that tell the cell when to divide. It's an elegant chain of command. If the environment changes, say temperature goes up, the cell might respond by turning on genes for heat shock proteins. If a hormone signal comes, it might alter the expression of certain genes. It's dynamic yet also stable. Each cell type remembers which genes to keep active for its function. In our meditative state, think of the DNA in each of your cells as a very long, gentle melody. The bases A, T, C, G are like musical notes. Different segments, genes, are like different motifs in the music. When a cell expresses a particular motif, it produces a certain effect, protein, which contributes to the symphony of your body's functions. Sometimes the music is loud, a gene highly expressed, sometimes soft, low expression, and sometimes silent, gene off. And your cells have been playing these tunes correctly since you were a single-cell zygote, all thanks to the reliable replication and expression of DNA. It's quite magical. Having delved into the molecular underpinnings, we will now scale up again to consider something emergent from all these cellular activities. How does an entire organism maintain balance? How do cells talk to each other? How are things like temperature, hydration, and energy levels kept steady in a body? This brings us to homeostasis and the endocrine and nervous systems, which coordinate bodily functions. We'll maintain our gentle pace as we see how the body self-regulates and communicates internally, linking the micro-world of cells to the macro-world of tissues, organs, and organ systems. Let's proceed calmly into that realm, knowing that everything we discuss still ultimately relies on the molecular biology we've covered, but we can appreciate it on a higher level now. Homeostasis, balance in the body life, especially for complex organisms like us, is a balancing act. Our internal environment, things like temperature, pH, water content, iron levels, blood sugar, etc., needs to stay within certain ranges for our cells to function optimally. Homeostasis is the term for this dynamic equilibrium, the steady state that organisms maintain internally even as external conditions fluctuate. It's like the body's way of staying calm and stable inside, no matter if outside it's hot or cold, or if you've eaten a meal or are fasting. A classic example of homeostasis is body temperature in humans. We are warm-blooded, endothermic, maintaining about 37 degrees Celsius, 98.6 Fares core temperature. If you go out on a cold day, your body doesn't just drop to the surrounding temperature. Instead, mechanisms kick in to keep you warm. You might start shivering, small muscle contractions that generate heat. Blood vessels in your skin may constrict, narrow, to reduce blood flow near the surface, conserving heat for vital organs. If you get too hot, the opposite happens. You sweat, And as sweat evaporates, it cools you, and blood vessels in skin dilate to release heat while you get flushed. These are homeostatic responses, actions triggered to oppose the change and bring the body back to the set point. This is an example of negative feedback, the cornerstone of many homeostatic mechanisms. Negative feedback means that if a change occurs, like temperature rising, the body triggers responses that counteract that change, cool the body down, thus reversing the trend and bringing conditions back towards the target. Let's break down negative feedback in a gentle scenario. Say you are resting under blankets and your body warms slightly above normal. Temperature sensors, thermoreceptors in your skin and brain detect this and send signals to your brain's thermostat region, the hypothalamus. The hypothalamus in turn orchestrates a response. It may cause you to sweat and breathe a bit faster. Panting slightly can also release heat and tell your blood vessels to widen. These actions gradually cool you. Once your temperature falls to the normal range, the sensors note it and the hypothalamus turns off the cooling responses. The term negative feedback comes because the effect, cooling, negates or reduces the original stimulus, overheating. Similarly, if you were cold, the effect, warming by shivering, negates the stimulus, too cold. This ensures stability. Most homeostatic controls are of this negative feedback type. Another important one is blood glucose regulation. When you eat and digest a meal rich in carbohydrates, glucose enters your bloodstream and blood sugar levels rise. Your pancreas detects this and secretes insulin, a hormone. Insulin acts like a key allowing glucose to enter cells, especially muscle and fat cells, and also signals the liver to take up glucose and store it as glycogen. The result is that blood glucose levels fall back to normal as the glucose moves out of the blood into storage or usage. That's negative feedback. High glucose triggered insulin, which lowers glucose, removing the initial trigger. Later, if you haven't eaten for a while and blood glucose drops too low, the pancreas releases another hormone called glucagon. Glucagon signals the liver to break down glycogen and release glucose back into the blood, raising the blood sugar level to normal. Insulin and glucagon are like gentle push-pull partners, keeping glucose around a happy medium. We'll talk more about them in the endocrine section, but as a homeostasis concept, it's clear. Two opposite hormones maintain the balance depending on the direction of change needed. Our bodies maintain homeostasis for so many things. pH of blood, around 7.4, using buffers like bicarbonate and adjusting breathing to retain or expel CO2, since CO2 can acidify blood. Blood pressure, sensors in arteries and adjustments via heart rate and vessel dilation. Water balance. If dehydrated, your brain makes you thirsty and the kidneys concentrate the urine to save water. If overhydrated, you produce more dilute urine. Ion balance, like sodium, potassium, calcium, regulated by hormones that affect kidney function or bone storage of minerals. And oxygen levels. If O2 is low, you breathe faster, or if chronic, the body may produce more red blood cells. An interesting aspect is the role of the nervous system and endocrine system in homeostasis. We have detectors, sensory nerves or specialized cells that monitor changes, control centers, often the brain or endocrine glands that decide what to do, and effectors, muscles, glands, organs that carry out the adjustments. For instance, for temperature, detectors in skin, control center in hypothalamus, effectors are sweat glands, muscles, blood vessels. For blood sugar, detectors and control center are in the pancreas. The beta cells sense high sugar and release insulin. Effectors are body cells and liver. We'll delve more into the coordination by hormones and nerves soon, but it's amazing how automatically all this happens. We usually don't have to think, oh, I'm too warm, I should start sweating now. It just happens thanks to our body's built-in control systems. There's also positive feedback in biology, but it's used more sparingly, usually to drive processes to completion rather than maintain stability. Positive feedback is when a change triggers further change in the same direction. A classic example is childbirth. Contractions of the uterus push the baby toward the birth canal. Stretch receptors detect this and signal the release of oxytocin hormone, which causes stronger contractions, which cause more stretch and thus more oxytocin, a snowball effect until the baby is delivered. Once the baby is out, the stimulus stretch stops and the loop ends. Another example is blood clotting. When a vessel is damaged, platelets start sticking to the site and release chemicals that attract more platelets and so on, quickly forming a clot. After the vessel is plugged, the feedback loop is halted. Positive feedback is more like an amplification to achieve a result, not a steady-state regulator. In our meditative context, think of homeostasis as the body's way of whispering to itself, gentle signals to tweak here, adjust there, ensuring everything inside remains in harmonious balance. You don't consciously direct your blood sugar or iron levels. It's like your body has its own wisdom and caretaking, a silent caretaker adjusting the thermostat, the hydration, the fuel mixture of your inner environment. We often notice homeostasis only when it's being challenged. For example, during exercise, you sweat and breathe harder, heart rate goes up. Those are homeostatic responses to keep core temperature safe and deliver more oxygen to muscles, plus remove CO2. When you have an infection, you might get a fever. Interestingly, that's the body raising the set point for temperature to help fight microbes, temporarily altering the homeostasis for a purpose. If homeostasis fails significantly, that's when trouble arises, like in diabetes, where insulin regulation of glucose is impaired, leading to chronically high blood sugar, which can damage organs. Homeostasis applies not only to the whole body, but within cells too. Each cell regulates its internal environment, for instance, keeping a certain concentration of ions like calcium or sodium, adjusting its metabolic reactions according to need. But the big picture in multicellular organisms is that organs and systems work together to maintain the environment in which all cells live, the extracellular fluid. Now that we have an understanding of homeostasis, let's explore two critical systems that help maintain it: the endocrine system and the nervous system. The endocrine system uses chemical messengers, hormones sent through the bloodstream to distant organs, signaling them to adjust, like insulin and glucagon as we discussed. The nervous system uses electrical impulses and neurotransmitters for rapid communication, ideal for quick and targeted actions like moving your hand away from a hot stove or adjusting heart rate on a moment-to-moment basis. We'll first talk about the endocrine system in our gentle narrative, meeting some of the major hormones and glands, and then we'll move to the nervous system, looking at how nerves transmit signals in a calm yet astonishing way. Both systems beautifully illustrate the body's internal harmony and how different parts of the body talk to each other to keep the whole in balance. So take a deep breath and appreciate how even that act, breathing, is part of homeostasis, maintaining oxygen and carbon dioxide levels, regulated by your brainstem automatically. As you exhale, let's proceed to quietly examine the hormones and signals that sustain life's equilibrium. The endocrine system, gentle chemical messengers. The endocrine system is like the body's slow, steady communication network, using the bloodstream to send chemical messages called hormones. While the nervous system is rapid and specific, like a text message to a particular person, the endocrine system is generally slower and more broadcast, like a gentle radio signal that only certain receivers pick up. Hormones help regulate numerous body functions: growth, metabolism, reproduction, blood pressure, and many aspects of homeostasis, such as blood sugar, stress responses, and more. Endocrine glands are ductless glands that release hormones directly into the blood. Let's imagine and stroll through some of the major endocrine glands and their hormones, keeping our tone soft and explanatory. Hypothalamus and pituitary. These are deep in the brain and often considered the master regulators. The hypothalamus is actually a part of the brain that links the nervous and endocrine systems. It monitors the body's state, temperature, blood osmolarity, etc., and secretes hormones that either stimulate or inhibit the pituitary gland, which lies just below it. The pituitary gland is often called the master gland because it releases hormones that regulate other endocrine glands. However, the pituitary takes many cues from the hypothalamus. The pituitary has two parts, anterior and posterior. The anterior pituitary produces hormones like TSH, thyroid-stimulating hormone, ACTH, adrenocorticotropic hormone, GH, growth hormone, prolactin, FSH and LH, which regulate the reproductive organs. Each of these travels via blood to target glands or tissues. For instance, TSH tells the thyroid gland to produce thyroid hormone, ACTH tells adrenal cortex to release cortisol, GH has widespread growth effects, etc. The posterior pituitary doesn't make hormones itself, but stores and releases hypothalamus-made hormones like ADH, antidiuretic hormone, and oxytocin. ADH, also called vasopressin, helps kidneys retain water, part of water balance. When you're dehydrated, ADH levels rise to conserve water. Oxytocin is famous for roles in childbirth, causing uterine contractions via positive feedback, and milk ejection during breastfeeding, and also is linked to social bonding. Thyroid gland. This is a butterfly-shaped gland in your neck around the trachea. It produces thyroid hormones T3 and T4 when stimulated by TSH from the pituitary. Thyroid hormones set the metabolic rate of the body, like a dial for how fast cells burn energy. If there's lots of thyroid hormone, metabolism revs up, you feel warm, maybe lose weight, heart beats faster. Too little and metabolism is sluggish, fatigue, feeling cold, weight gain. Thyroid hormone is crucial for development too, especially of the nervous system in infants. The thyroid also produces calcitonin, a hormone that can lower blood calcium by depositing calcium in bones, but in humans it's not a major regulator, more so in other animals. The counterpart to calcitonin is parathyroid hormone, PTH, from the parathyroid glands, tiny glands on the back of the thyroid. PTH raises blood calcium by breaking down bone, releasing calcium and increasing calcium absorption in gut and kidneys. Calcium levels are tightly controlled because calcium is vital for muscle contraction, nerve function, and more. Pancreas. We already discussed its key hormones. The pancreas has endocrine clusters, islets of Langerhans, that release insulin from beta cells and glucagon from alpha cells. Insulin lowers blood glucose by facilitating its uptake into cells. Glucagon raises blood glucose by stimulating the liver to release stored glucose. The balance of these hormones keeps blood sugar stable. In diabetes mellitus, either insulin production is insufficient, type 1, often because the immune system destroyed the beta cells, or cells don't respond well to insulin, type 2, often linked to insulin resistance. The result is chronically high blood sugar, which can cause various complications if not managed. Adrenal glands. These sit atop each kidney. They have two parts, adrenal cortex, outer, and adrenal medulla, inner, and they produce different sets of hormones. The adrenal medulla is part of the sympathetic nervous system and secretes epinephrine, adrenaline, and norepinephrine during stress, fight or flight response. These cause an increase in heart rate, blood pressure, blood flow to muscles, and blood sugar for quick energy, preparing the body to handle an emergency or strenuous activity. The effect is swift but relatively short-lived, as these are neural stimuli. The adrenal cortex produces steroid hormones, including cortisol and aldosterone. Cortisol is a glucocorticoid that helps the body respond to long-term stress and has effects on metabolism, raises blood sugar by promoting gluconeogenesis, etc. It also dampens the immune system, which is why synthetic versions are used as anti-inflammatory drugs. Cortisol's release is triggered by ACTH from the pituitary. Aldosterone is a mineralocorticoid that helps regulate blood pressure by instructing kidneys to retain sodium and thus water and excrete potassium. More sodium and water retention raises blood volume and blood pressure. Aldosterone release is regulated by the renin-angiotensin system in response to low blood pressure or low sodium. Gonads, ovaries and testes, these produce sex hormones. Testes produce testosterone, which promotes male secondary sexual characteristics like facial hair, deeper voice, and is necessary for sperm production. Ovaries produce estrogens and progesterone. Estrogens, like estradiol, promote female secondary sexual characteristics like breast development and help regulate the menstrual cycle, among many roles. Progesterone is key in preparing the uterus for pregnancy and maintaining early pregnancy. The release of these sex hormones is under control of the pituitary hormones SH and LH, follicle-stimulating hormone and luteinizing hormone, in both sexes, part of a feedback loop. For instance, high levels of estrogen or testosterone can feed back to reduce FSH-LH release. Other hormone producers. There are many other hormones and organs with endocrine functions. The pineal gland in the brain secretes melatonin, which helps regulate sleep-wake cycles. circadian rhythms. The thymus, active in children, produces thymosin, which is involved in immune T-cell development. The thymus shrinks as we age. The kidneys produce erythropoietin, EPO, a hormone that stimulates red blood cell production when oxygen is low. The heart produces AMP, atrial natriuretic peptide, when blood pressure is high, which basically opposes aldosterone. It causes kidneys to excrete more sodium and water to reduce blood volume. The digestive tract has hormones like gastrin, secretin, etc. that regulate digestion. Fat tissue even produces hormones like leptin, involved in appetite regulation. In a restful story sense, imagine these glands as different players in an orchestra, each releasing its own melody, hormone that travels through the blood, the air of the concert hall, to reach target instruments, organs that will then adjust their tune accordingly. The hypothalamus and pituitary might be the conductors, ensuring everything stays in harmony. The endocrine signals are slower than nerve signals. It can take seconds to minutes for hormones to circulate and cause effects, but their influence can last minutes to hours or even longer. This is suitable for functions that aren't instant emergencies, growing, metabolizing, adjusting to daily cycles, etc. For instance, if you're frightened, your adrenal medulla will release adrenaline within seconds to ready your body. But for regular metabolism, your thyroid hormone levels adjust gradually and keep cells' metabolic pace steady. Growth hormone spikes a bit at night to help with tissue maintenance. These are rhythmic or homeostatic tunes. One can appreciate how endocrine disorders can upset the balance. Too much thyroid hormone, hypothyroidism, and everything speeds up, leading to weight loss, anxiety, rapid heartbeat. Too little hypothyroidism and things slow down, causing fatigue, weight gain, cold sensitivity. Or in diabetes, lack of insulin action means blood sugar goes way out of tune, affecting many body systems. Fortunately, many of these can be managed nowadays, like synthetic hormones or medications. Now we have covered how the body uses chemical messages to maintain steady functions. Next, we will explore the nervous system, the body's fast communication network that coordinates actions and reactions with breathtaking speed and specificity. While hormones are like mailing letters or broadcasting radio signals, nerve impulses are like instant phone calls or direct electrical wires connecting parts. Keep relaxed as we move into how neurons transmit impulses, how the brain integrates information, and how we respond to the world. Despite the complexity, we'll keep descriptions simple and calm. The nervous system, the body's electrical network. The nervous system is the body's rapid communication and control system. It uses electrical signals, nerve impulses, and chemical signals, neurotransmitters, to send messages from one part of the body to another almost instantly. Through the nervous system, we perceive the world, senses, think, form memories, make decisions, and coordinate all voluntary and many involuntary actions. Let's break it down gently. The fundamental unit of the nervous system is the neuron, or nerve cell. Neurons are specialized cells that can generate and transmit electrical impulses. They have a unique shape, typically a cell body, soma, which contains the nucleus and metabolic machinery, many dendrites, branch-like extensions that receive signals from other neurons, and a single long axon, which carries the signal outward to other neurons or muscles. At the end of the axon, it usually branches into terminals that form connections, synapses, with the next cell. Neurons communicate at junctions called synapses. When an electrical impulse, action potential, travels down an axon to the end, it triggers the release of a chemical messenger called a neurotransmitter into the small gap, synaptic cleft, between that neuron and the next cell. The neurotransmitter diffuses across the cleft and binds to receptors on the next cell, which could be another neuron or a muscle cell or gland cell, influencing it, maybe exciting it to fire its own impulse, or inhibiting it from firing. Then the neurotransmitter is quickly removed or degraded, so the effect is brief. This is how signals can be passed neuron to neuron in circuits. We have billions of neurons in our brain alone, forming a complex network, along with supporting glial cells, which nourish, insulate, and protect neurons. How do neurons generate an electrical impulse? This involves ions, charged atoms like sodium, potassium chloride, Cl, Neurons maintain a voltage difference across their membrane. Inside is negative relative to outside, typically about -70 millivolts at rest. This is called the resting potential, maintained by pumps that keep more Na outside and more K inside, plus other charged molecules. When a neuron is stimulated, say by a neurotransmitter binding at a dendrite, if the stimulus is strong enough, it causes sodium channels to open and NR rushes into the cell because of concentration and electrical gradient, making the inside less negative, depolarization. If the depolarization reaches a threshold, it triggers an action potential, a brief reversal of membrane charge that travels along the axon like a wave. Essentially, more Na channels open along the axon in sequence, propagating the impulse, and then K-Me channels open to let K out, repolarizing, restoring the negative inside. This wave travels fast in myelinated axons wrapped in insulating glial membrane segments called myelin. It can reach speeds up to 100 meters a second, over 200 miles per hour. Myelin sheaths have gaps, nodes of Ranvier, where the action potential jumps from node to node, speeding conduction, saltatory conduction. Between signals, the neuron resets ion gradients via pumps, ready to fire again. Now, the central nervous system, CNS, consists of the brain and spinal cord. It's the processing center, integrating sensory information, making decisions, consciously or reflexively, and sending out commands. The peripheral nervous system, PNS, includes all the nerves outside the CNS that connect to limbs and organs. Within PNS, we have sensory, afferent, neurons bringing info in, like from skin, eyes, ears to CNS, and motor, efferent neurons carrying signals out to effectors like muscles or glands. Motor division splits into somatic, voluntary control of skeletal muscles, and autonomic, involuntary control of smooth muscles, heart, glands. The autonomic system further has sympathetic and parasympathetic divisions. Sympathetic fight or flight responses, for example, it increases heart rate, dilates airways, releases adrenaline, etc. to handle stress. And parasympathetic, rest and digest, it slows heart rate, stimulates digestion, etc., promoting calm and maintenance activities. These two act like yin and yang, balancing body functions unconsciously. Think of touching something hot. Sensory receptors in your skin detect the high temperature and pain, sending an impulse via sensory neurons to your spinal cord. Their interneurons process it, and immediately, before you even think, a motor command is sent back via motor neurons to your arm muscles to contract and pull away. This is a reflex arc, a protective mechanism that's handled at the spinal cord level for speed. Only after that do you feel the pain consciously, as the signal also goes to your brain. Reflexes like this show how the nervous system can trigger appropriate responses almost automatically. The brain itself is incredibly complex, but we can mention some parts gently. The cerebrum is the largest part, divided into two hemispheres responsible for higher functions like reasoning, imagination, and voluntary movement control. It has regions, lobes, specialized for vision, hearing, touch sensation, language, etc. The cerebellum, at the back below cerebrum, coordinates movement and balance, letting you perform smooth motions. The brainstem, midbrain, pons, medulla, controls vital functions like heart rate, breathing rhythm, sleeping, and a relay for signals. The limbic system deep inside deals with emotions and memory, including the hippocampus for memory formation and amygdala for fear emotion. The brain is like an orchestra of billions of neurons with trillions of connections, synapses, but remarkably, patterns of electrical and chemical signals across these networks correspond to our thoughts, feelings, and skills. The nervous system's function in homeostasis is through rapid adjustments. For example, if your blood pressure drops suddenly, like standing up quickly, baroreceptor sensors in arteries signal the brainstem, which, via the sympathetic nervous system, causes the heart to beat faster and vessels to constrict, raising blood pressure back up. It's a quick neural reflex. Or when you see something in the road while driving, you react almost instantly by hitting the brakes. That's the brain processing visual info and sending motor commands in fractions of a second. We should also mention neurotransmitters, these chemicals at synapses. There are excitatory ones like glutamate, primary excitatory in brain, and acetylcholine, important at neuromuscular junctions and in parasympathetic system, and inhibitory ones like GABA, primary inhibitory in brain, and glycine in spinal cord. There are modulatory ones like dopamine, involved in reward and movement. Deficit in dopamine in certain brain areas causes Parkinson's disease. Serotonin, mood, sleep. Norepinephrine, attention, part of sympathetic responses. And many others, over 100 types, including endorphins, which reduce pain and induce euphoria, etc. Many psychoactive drugs or medications work by altering neurotransmitter levels or receptor binding, like SSRIs for depression increase serotonin at synapses, or anesthetics might block pain neurotransmission, etc. In a calm narrative context, imagine your nervous system as a network of glowing wires and twinkling synapses. When you decide to move your hand, a thought in your motor cortex, in cerebrum, triggers signals that travel down your spinal cord and out to your hand muscles, all in the blink of an eye. When you listen to soft music, sound waves trigger signals in your ear's neurons, which travel to your brain's auditory centers, where they are perceived as melody. All these signals coalesce seamlessly into experience. The nervous and endocrine systems often work together. For instance, in stress, the sympathetic nervous system triggers immediate responses, heart race, etc., and also signals the adrenal medulla to dump adrenaline for a longer effect. Meanwhile, the brain's hypothalamus signals the pituitary to eventually release ACTH and cortisol for a more prolonged effect. In reproduction, brain signals trigger pituitary hormones which affect gonads, yet sexual function also involves nerves, like arousal and climax involve nerve signals. So the body doesn't treat these systems as isolated, they interconnect richly. We have come a long way from atoms to human organ systems. We can appreciate now how our body, at rest, is a concert of soft signals, hormones drifting through blood, nerves sending tiny impulses, all to keep our internal environment steady and to allow us to sense and interact with the world. It's remarkable that all this happens mostly without our conscious awareness. It frees us to focus our mind on other things or to relax and fall asleep, confident that our heart will keep beating and our lungs breathing under the watch of our autonomic nervous system and brainstem, and that our endocrine system will keep our blood sugar and salts balanced until morning. Finally, before we wind down, we will briefly turn to the realm of genetic tools and techniques. Essentially how humans have learned to explore and manipulate the molecular genetics we discussed for purposes like medicine, forensics, and research. We'll keep it broad and simple, then conclude our journey and drift into a final, relaxing summary. Genetic tools and techniques, exploring the code of life. In the last few decades, scientists have developed ingenious tools to study and manipulate DNA, the genetic material. These techniques allow us to diagnose diseases, clone genes, make medicines like insulin, and even edit genomes. While delving deep into each is complex, we will calmly survey some of the key tools and what they do in simple terms. DNA extraction and recombinant DNA. One of the fundamental things scientists learned was how to extract DNA from cells and then cut and paste DNA sequences. Special enzymes known as restriction enzymes act like molecular scissors that recognize specific short sequences in DNA and cut at those sites. For example, one might cut at GAT-GAC. Each enzyme has its own signature cut sequence. These cuts often create sticky ends, overhanging single-stranded DNA, which can pair with complementary sequences. Then, if we have another piece of DNA cut with the same enzyme, their sticky ends can match up and be joined by another enzyme called DNA ligase. This way, we can take a gene from one organism and insert it into the DNA of another, making recombinant DNA. For instance, the human insulin gene can be inserted into a small circular DNA called a plasmid, which is like a mini chromosome often found in bacteria. That recombinant plasmid can be introduced into bacteria, a process called transformation, and then the bacteria will use that gene to make human insulin protein, which can be purified for treating diabetes. This was a breakthrough, producing insulin without needing animal pancreases, and is one of many examples, other growth hormone, clotting factors, etc., made by bacteria or yeast engineered with human genes. This whole area is genetic engineering or biotechnology. Polymerase chain reaction. This is a technique to make millions of copies of a specific DNA segment, sort of an amplification method. It's like photocopying a certain page from a huge book until you have a big stack of that page. PCR uses a heat-stable DNA polymerase, nucleotides, and short primers that flank the region of interest. By cycling temperature, DNA is denatured, strands separated, primers anneal to target sequences, and polymerase extends to copy that region. Repeating this exponentially amplifies the DNA fragment. PCR is extremely useful. For example, in forensic science, tiny DNA from a crime scene, like a drop of blood or a hair, can be PCR amplified to have enough to analyze. Or in medical diagnostics, PCR can detect the DNA, RNA of pathogens, like viruses, in a patient sample, even if present in low amount. It's sensitive and quick. Amplification can be done in hours. DNA sequencing. Determining the sequence of nucleotides, A, C, G, in a DNA sample is sequencing. The classic method, Sanger sequencing, uses special chain-terminating nucleotides to create fragments that end at each position, which when run on a gel can be read to reveal the sequence. Modern sequencers are far faster and can sequence whole genomes in days. Sequencing has allowed us to read the entire human genome, all 3 billion letters, and the genomes of many organisms. This opens understanding of genes, evolution, and can identify mutations that cause diseases. Gel electrophoresis. This is a lab method to separate DNA fragments or proteins by size. DNA samples are loaded into a gel, like a slab of jello, and an electric current is applied. DNA is negatively charged due to phosphate backbone, so it moves toward the positive electrode. Smaller fragments move faster further through the gel pores than larger ones. After running, you stain the gel and see bands. Each band is DNA of a certain size. This can be used to visualize PCR results, check if a restriction enzyme cut worked, the DNA will be one band to two smaller bands if cut, or to do DNA fingerprinting. DNA fingerprinting is a use of electrophoresis plus PCR of regions that vary between people, like short tandem repeats in the genome. Because everyone, except identical twins, has a unique pattern of these repeats, analyzing several of them can identify a person from a sample. This is used in forensics, matching crime scene DNA to suspect paternity tests, etc. Southern, northern, western blots. These are techniques to trans DNA, southern, RNA, northern, or protein western, from a gel to a membrane, and then detect specific sequences or proteins with labeled probes or antibodies. For example, a southern blot can check if a person's DNA has a specific gene or mutated segment by using a complementary DNA probe. Western blot uses antibodies to detect specific proteins, commonly used to verify the presence of a protein, say HIV proteins in a sample as a test. Cloning. In molecular terms, cloning a gene means making copies of it, often via recombinant DNA in bacteria. But whole organism cloning is creating a genetic copy of an organism. The famous example was Dolly the sheep in 1996, the first mammal cloned from an adult cell. This was done by taking a nucleus from an adult cell and putting it into an egg cell that had its nucleus removed, then stimulating it to develop into an embryo and implanting into a surrogate mother. Dolly was genetically identical to the sheep that donated the nucleus. Cloning is still inefficient and controversial for higher animals, but it's shown that differentiated cells still have all DNA needed to form a whole organism, genomic equivalence. CRISPR-Cas9, a relatively new, powerful gene editing tool derived from a bacterial immune system. It uses a guide RNA to target a specific DNA sequence and an enzyme, Cas9, to cut there. This can knock out a gene or allow insertion of a new sequence. It's much more precise and easier than previous gene editing methods. Scientists are researching CRISPR for fixing genetic diseases by editing patient cells, but it also raises ethical considerations like the idea of editing embryos or germline cells that would pass changes to future generations. Gene therapy. The attempt to treat diseases is by delivering a correct gene to patients who have a faulty one. For example, some children are born with SIAD, severe combined immunodeficiency, due to a single gene defect. Some gene therapy trials have used modified viruses to insert a normal gene into their bone marrow cells to cure the condition. There have been successes and setbacks. Some early trials caused leukemia due to insertional mutagenesis. The new gene landed in a bad spot in DNA, but gene therapy holds promise for many genetic disorders if delivered safely. PCR in diagnostics and research. We mentioned PCR for detecting viruses. Another use is in COVID-19 testing. An RT-PCR test, reverse transcription PCR, can detect the RNA of SARS-CoV-2 virus in a swab sample, converting RNA to DNA, then amplifying it. If it amplifies and is detected, the sample had viral RNA, meaning infection. PCR is also used to amplify ancient DNA, like from a Neanderthal bone, for study, or to amplify traces of DNA in environmental samples. Bioinformatics. With all the sequencing data, a whole field of computer analysis emerged to handle and interpret DNA protein data. Scientists can compare gene sequences between species, predict protein structures, and even use CRISPR designing tools via bioinformatics. For a meditative perspective, consider how far we've come. We learned nature's secrets, that all life's complexity is encoded in sequences of DNA, and now we can read and even rewrite those sequences. It's as if for millennia we appreciated the beauty of a symphony, life by ear, and now we've obtained the musical score and even the instruments to modify the music. It's powerful and must be used carefully and ethically. These genetic tools are aiding in creating treatments like manufacturing insulin, as mentioned, or monoclonal antibodies for therapy, improving crops, GMOs that are pest-resistant or more nutritious, and understanding our ancestry and evolution, like sequencing genomes of various humans and finding how closely we are all related or our relation to other hominids. As with any technology, there's a balance to be struck, but overall, these tools expand our ability to care for life and understand it. Now, having traversed through fundamental chemistry, cellular biology, physiology, and biotechnology, let's start winding down. We have covered a broad curriculum of biological knowledge in a calm, story-like manner. It is time to slowly transition to rest, summarizing gently what we have learned, and then drifting off, appreciating the wonders of life from the microscopic to the whole organism. You've done a wonderful job following along this extensive journey. In the next section, we will softly recap our major topics, reinforcing the key ideas in a soothing, lulling way, and then conclude our scientific bedtime story, allowing you to embrace sleep with perhaps a bit more knowledge embedded in your dreams. Winding down, a gentle recap and descent into sleep. We have journeyed far and wide across the landscape of biology in a calm and unhurried way. Now, as we prepare to rest, let's softly revisit the highlights of our exploration, allowing the concepts to settle pleasantly in the mind without any urgency. Think back to the beginning, where we started with the smallest pieces, atoms bonding to form molecules. We remember that water is special, binding gently with hydrogen bonds, making life possible. We recall that carbon is the versatile backbone of life, capable of forming 4 bonds and thus a diversity of structures. Life's chemistry taught us about acids and bases balancing pH and buffers quietly keeping that balance. All these chemical fundamentals set the stage for more complex structures. We then built up to the macromolecules of life, the sweet and sustaining carbohydrates, the energy-rich and membrane-forming lipids, the hard-working and versatile proteins, and the information-storing nucleic acids. In our minds, we see a chain of glucose units forming starch in a seed, a droplet of oil representing lipids concentrated in energy, a folded enzyme protein speeding a reaction, and the elegant double helix of DNA quietly holding genetic instructions. Each type of macromolecule plays its role, and together they compose the structures and engines of cells. We moved into the cell, the tiny living unit, and imagined its parts. The protective cell membrane fluid and alive, the mitochondria breathing quietly to supply ATP, the ribosomes diligently assembling proteins, the nucleus safeguarding DNA, and softly allowing transcripts, mRNA, to exit through its pores. We saw how cells maintain an internal order, how substances move across membranes, sometimes with ease, diffusion, other times with care, active transport, and how cells communicate with neighbors through gentle signals and junctions, forming tissues and organs in a multicellular chorus. We delved into the energy pathways, how cells derive energy from food through cellular respiration, a graceful breakdown of glucose with oxygen, yielding water, carbon dioxide, and that precious ATP energy. We recalled glycolysis splitting sugar, the Krebs cycle spinning away, and the electron transport chain humming in the mitochondrial membrane. It was a complex dance, yet we boiled it down to a simple truth. We breathe in to support our mitochondria. We breathe out what they produce, and we eat to give them fuel. Then we turn to photosynthesis in plants, the mirror image process where chloroplasts in leaves quietly capture sunlight and breathe in carbon dioxide to weave it into sugars, releasing oxygen for us to breathe. We imagined sunlight filtering through a leaf, energizing electrons in chlorophyll, splitting water, and ultimately producing glucose and oxygen, a gentle gift of nourishment and air from the plant world to us. Photosynthesis and respiration, two halves of a whole, cycling carbon, oxygen, and energy between organisms in a balanced global rhythm. Our journey then took us into the genetic code and how it's used. We relaxed into the understanding that DNA, in its quiet double helix, holds a library of recipes for proteins. We listened to the soft scratch of transcription, DNA's script being copied into mRNA, and the steady tapping of translation, ribosomes reading mRNA codons and stringing amino acids into proteins. It's amazing how such a tiny code can orchestrate the building of an entire organism. We also gently touched on how cells divide and DNA replicates, so each new cell has a complete library. Through it all, we kept our tone calm, realizing these processes have gone on in every cell trillions of times, even as we rest or sleep without any need for conscious control. We surveyed the physiology of homeostasis, seeing our body as a wise caretaker that keeps internal conditions stable. We felt gratitude for our body's automatic balancing acts, maintaining temperature so we're not too hot or cold, adjusting our breathing and heart rate, keeping our blood chemistry in check. We noted negative feedback loops, like a thermostat analogy, detecting a deviation and gently correcting it. If our blood sugar rose after a sweet snack, insulin came to the rescue to lower it. If it fell because we haven't eaten, glucagon nudged it back up. We can appreciate these subtle interventions that happen throughout the day and night, keeping us alive and well without disturbing our consciousness. And we remembered that sometimes a positive feedback loop takes over, like in childbirth, but that too has its own time and purpose, resolving when the job is done. Then we quietly walked through the endocrine garden, meeting the glands that secrete hormones. We imagined the pituitary in the brain as a master switchboard, sending signals like TSH, ACTH, growth hormone, and more to regulate other glands. We saw the thyroid like a butterfly on the neck, releasing hormones that set our body's metabolic tempo. We pictured the adrenals atop the kidneys, handing out stress hormones, a burst of adrenaline in an emergency, a drizzle of cortisol during prolonged stress, and how they help us adapt. The pancreas, with its insulin and glucagon, we recognized as a key player in energy balance. The sex glands releasing estrogen, progesterone, testosterone, guiding development and reproduction. All these hormones circulating in the blood, each finding their target cells to whisper instructions. It's like a slow, soothing communication system working in parallel to the quick chatter of nerves. Speaking of the quick chatter, we journeyed into the nervous system, marveling at the swift electricity of neurons. We saw how a thought or sensation travels as an impulse down an axon, a flicker of voltage, and jumps a synapse via neurotransmitters to excite the next cell. We imagined the brain with billions of such flickers, somehow weaving the symphony of consciousness, and we did so in a calm, non-technical way, just appreciating the wonder. The spinal cord's reflex arcs gave us a sense of security, that some responses are reflexive and protective without needing our input. We felt how the autonomic nerves keep our heart and digestion on track. The sympathetic system acting as the accelerator when needed, the parasympathetic as the brake and restorer, all of it so well coordinated, like a serene but capable guardian always on duty. Finally, we touched on the tools humans have developed to peek at and even modify biology's secrets. We mentioned PCR as a way to amplify DNA. How amazing that we can take a minute amount of genetic material and duplicate it so many times for study. We talked about recombinant DNA and how human insulin is produced by bacteria thanks to gene cloning, benefiting millions of people. We skimmed over sequencing, CRISPR, gene therapy, all the ways we are learning to read and write the language of DNA. It reminds us of the dual nature of knowledge. It empowers us to heal, feed, and understand, and it also asks us to be wise stewards of that power. But for tonight, it's enough to know that the code of life has been deciphered to a large extent, and that knowledge itself stands as a tribute to human curiosity and ingenuity. Now, with these recollections made, let's allow ourselves to gently let go of the details. There is no need to actively remember or analyze now. Trust that your mind has absorbed what it needed and that during sleep it will quietly file away any new knowledge in your memory, just as cells file away experiences or adapt during rest. Indeed, sleep helps consolidate learning, even in biology class. Feel the calmness that comes from seeing the coherence in life's processes. How, from atoms to cells to entire organisms, there is an underlying order and balance. And feel the calmness in your own body right now. Your breathing is steady, your heart is at rest, your body temperature comfortable, all thanks to the wonderful biology within you working harmoniously. You can release any remaining wakeful thoughts knowing that your body and brain are unwinding into the gentle rhythm of sleep. As you lie here, perhaps you can visualize something soothing in the realm of biology, maybe a soft green leaf in sunlight, performing photosynthesis quietly, or an ocean wave reminiscent of the waves of impulse that travel down neurons, or a slow heartbeat echoing the life force in all animals. Choose whatever image makes you feel most at peace and safe. You might imagine yourself floating through the bloodstream on a tiny raft, carried effortlessly by the flow, passing by red blood cells who kindly deliver oxygen, drifting by liver cells doing their metabolic work calmly, perhaps waving at them in gratitude for their tireless service. Or picture walking through a giant forest where each tree is like a cell, and you are small. You hear the gentle wind, which is like the whisper of hormones in the air, carrying signals, and the sunlight trickling down as knowledge or nourishment. All is well in this internal forest. Your mind can let these analogies go now too, as sleep nears. You've nourished your mind with science and your body with relaxation. The 2 hours of journeying through biology have not been in vain. Even if sleep took you at some point, your subconscious perhaps enjoyed the soft lecture, like seeds planted in a quiet garden. Feel free to release any effort of thinking now. You are free to simply be in this moment, a living organism at rest, much like countless others in the natural world who sleep at night. The flowers closing, the birds roosting under the same stars and part of the same biological rhythms. Your breathing is slow and soft. Your heart is calm. Each part of your body is relaxed from your toes to the top of your head. Within each of those parts, cells are gently carrying out their tasks, but they too may be in a sort of restful mode, doing maintenance and repair as you sleep. Trust that they have it under control. You do not need to supervise your biology. It functions best when you surrender to rest. It's time now to let go of the day and of this story. Time to drift into a deep, comfortable sleep, where your mind can wander through dream landscapes, perhaps even more wondrous than the cells and stars. Thank you for joining this gentle journey through biology. May it bring you not only knowledge, but also peace. Now, sink deeper into your pillow, feel the weight of your body supported fully, and allow the final quietness to envelop you. The scientific narration has ended, and only tranquility remains. Good night.